Today we're going to go over the main portion of neuro. This is a little bit extensive in number of slides to review. The nice part about it is most of it is familiar. I've already opened mine up, so I'm just going to switch screens. But as you start going through the beginning, the basic foundation of what we kind of do in this class is we go back and we look at things that we already have some knowledge about. Like, for example, the basic role of the nervous system was meant to be a communication coordination system, and it was meant to respond to stimulus. And we kind of build from those foundations by talking about all the parts and how they work together. So, for example, if I pick up some type of sensory information, it's carried or gets to some type of central processing unit, perhaps, maybe a portion of the brain that picks up touch. Maybe it's a portion of the brain that picks up temperature. And then we can properly respond to that stimulus in order to maintain homeostasis. So this basic example it gives on this early page is if I want to maintain a body core temperature somewhere between 98.1 and 99.1, I need to A, sense that information, B, set that information. Like if we want to hover around 98.6, give or take about a half a degree. And then we need to be able to respond to the input we're receiving. So for example, if I have a sensory receptor that picks up temperature, like a thermoceptor, and it's telling me that I'm cold, what would my response be? Maybe shiver. So I need to be able to connect back out to the body in order to control those things called effectors. So again, this isn't anything new that we're talking about yet. And a lot of this PowerPoint is going to function in this way where it's mostly a review of things you already know. But the key point now in this particular course is figuring out where failures can occur. So if I have maybe a demyelinating disease that's not allowing me to transmit signals, if I have some type of receptor-based disease, it's not allowing me to communicate, the signal is transmitted, those are going to cause problems. So the nervous system, there's a lot of places where problems can take place. So I'm sure you've already kind of opened the PowerPoint or not. And you've seen, again, how extensive it is. But I assure you, much of this stuff's going to come back easily. Most of it's pretty simple ideas. And basic roles of the nervous system, it acts as a communication coordination system. The basic role that it plays is being able to respond to stimulus. So again, how can I do that? Well, I have to be able to receive stimulus, process that information, and then control things like effectors. So as we go through, I'm kind of looking at this for the first time with you. I'm making quick judgment calls, time on a certain page, or I want to glance over it. But probably pretty early on, what it'll do is it gives you different varieties or ways in which people describe the nervous system. And because it's so complicated, a lot of different people have come along and says, well, if we subdivide it this way, I think it would be best. So some people do that structurally. Other people do that functionally. There's a real right way to do it. So this structure organization or structure organization of the nervous system is you kind of take the core, the processing units like the brain and the cord, and then you connect it to the rest of the body. So that's where things like CNS and PNS comes from. Again, the PNS, if you remember, is 12 pairs of cranial nerves. We'll review those in the next section, perhaps. For example, you go back, you look at schedule, fluid and electrolytes. This past week, here, Neuro, it's going to talk a lot of the components of the nervous system, different parts and pieces, what they do, how they work. Tracts gets mentioned again. Spinal cord cross section, I believe, gets mentioned again in this section. But then when it gets into the neurosensory, it goes back and it talks about like different types of information we can receive. And on one of these early slides in neuro, that's another way we subdivide the nervous system. Sometimes you subdivide it based off structural location. Sometimes you subdivide it based off function. So neurosensory, believe it or not, is the way we subdivide by function. And since special senses fall under that category, we'll kind of wait and review the cranial nerves when we do that. So for right now, one of the basic things is we just say there's 12 pairs of cranial nerves. So these are nerves that are directly coming off the brain, usually exiting the skull. Many of them are mixed, but not all. Some of them are strictly sensory, some of them are strictly motor, but some of them do both. So we'll talk about those later. And then when you get into the 31 pairs of spinal nerves, these are the ones that exit from the vertebral column or the spinal cord, and all of these would be mixed. They carry both sensory and motor information. And then that leads us to the next type of organization, which is functional organization, which more of the, I guess, emphasis is on what direction are we going? Are we going afferently? Are we going efferently? And afferent information tends to be all sensory in nature. Like I need to know what's going on in my environment, both internal environment and external environment. And then I need to be able to adapt to that appropriately. So if maybe my stomach is sending too much acid to my small intestines, which of course that happens all the time, we need to know about that because we don't want to burn the inner lining of the small intestines. So I need to know about that first, and then I need to be able to respond to it. So maybe we would communicate, in this case, picking up sensory information with nerves, but then send another signal using a hormone. And in that case, the hormone is going to tell us to send in some bicarbonate ion from the pancreas. So hormones, although not specifically part of the nervous system, function in a similar way where they are chemical communicators. But again, the basic concept, the basic idea is afferent information is telling you what's going on internally or externally. Like, again, acidity inside my vessels or in my digestive system, temperature inside and outside my body. Those are things we monitor all the time. And then we need to be able to respond appropriately. So when you do the efferent flow, the exiting from the CNS, that gets further subdivided into things that we can control voluntarily or volitionally, and then the things that we control through autonomics. And this ends up being a further discussion in which we talk about the autonomics, the endocrine together. And when I was in school, endocrine was always a part of the nervous system. Since your hypothalamus is basically running the endocrine system, it's kind of a further division. It's just the form of communication slightly different. So rather than using an axon terminal and a neurotransmitter that only has to cross that short gap, we can secrete a hormone into the blood, and then the blood will carry that chemical communicator over longer distances and maybe disperse it further. But it kind of functions in the same manner. So again, going back, so that's a general overview of not only what we're doing with this recording, but some of the information that's coming up and how that ties into just understanding the basics of these systems. If you like charts and graphs, same thing here. This is actually a pretty good picture. I've used this quite a few times in different, I guess, maybe your 101 course or 108 course. You would have seen this in chapter 10. No, you probably wouldn't have seen this until you got into cardio. My mistake. But I think that's the first time I showed you this picture where it was showing you where the cord itself was communicating with the sympathetic chain ganglia. And maybe we had talked about something called an accelerator nerve. So we're definitely going to come back and use this picture in the future. The blue being parasympathetic stimulus, the red being sympathetic stimulus. We'll talk about that. But again, this is just kind of showing you how things get connected. And one of the big overarching concepts that we're going to talk about is, well, what if this signal doesn't make it? How would that show up? And depending on where the signal gets blocked, whether it happens in here, or out here, would create a difference. So if I'm trying to do some type of, say, somatic motor response to something, maybe I'm trying to control a muscle, There's a difference in how that would look is if I had a lesion here or here, in which we would have what's called an upper motor neuron lesion, versus if we had a lesion somewhere out here. And again, I know these are kind of showing more autonomics, and that's not really how we control skeletal muscle, but it just gives you an idea of big picture ideas that we're looking at. Let's just make this clear for anybody that didn't have me previously. When you get into terminology associated with neurons, we do subclassify neurons both again structurally and functionally. But there's always this big emphasis on bringing up terms like axons and dendrites. And one thing I try to make clear in our Anatomy 1 class, our Biology 108 class, is dendrites would most appropriately be a term that we really only use for multipolar neurons. So when you talk about neurons, some of the most prevalent type of neurons that we have in the body are the multipolar neurons. So let me do a little sidebar on this real fast. And what I'll discuss is, again, classification of neurons, where these terms kind of are most appropriate to use. And that should cover a few of the slides for us. So again, there's classification of neurons based off function. Here's classification of neurons based off shape, where we find those things based off shape, and then where would be most appropriate to use terms like axons versus dendrites. So what I'm going to do, this is what I found to be easiest when I make recordings, is I'm actually going to get off my screen here, and you can see like a little box up in the corner. But this is just a little document camera, and I'm going to draw some pictures for you. And one of the most common pictures we draw when we talk about neurons, which are considered the functional units of the nervous system. So FXN is just a simple way of saying like function. So the functional units of the nervous system are again, the parts or the smallest pieces, the functional units are the smallest individual pieces of an overall structure or organizational unit that has the same basic functions that you would attribute with that larger unit. So for example, if my nervous system is known for communicating and coordinating or responding to stimulus, what are the smallest individual pieces that are responsible for doing that? They're called neurons. So neurons are not only a type of cell that we find in the nervous system, they're also considered the functional units of the nervous system. That's different than, say, glial cells, because glial cells, while they support neurons, they're not actively, or at least directly, involved with the responding to stimulus portion. So they might accelerate it, they might protect it, they might help clean up around it, but they're not the ones that are actually receiving the sensory information, carrying the motor information. They're just going to provide some type of supportive role. So again, neurons are what we consider the functional units of the nervous system, the small pieces. And then they get subdivided based off shape. So you end up having, say, like multipolar, you have bipolar, and then let's just go ahead and call the other one what it is rather than what it's sometimes referred to as, which is pseudo-unipolar. So pseudo-unipolar is the type that we actually have. Unipolar doesn't exist in humans. But multipolar is the ones that we talk about most frequently because it's the ones that make up the vast majority of the gray matter in the brain, the vast majority of the gray matter in the spinal cord. They're the ones that are supplying the axons that make up the white matter of the brain and make up the white matter of the spinal cord. So when I think about the CNS, I think about multipolar neurons because that's what's making up the majority of the things that we think about as our central nervous system. Bipolar neurons, these are going to be the ones that are going to be found usually around areas of special senses. That's where they're most famously located. So you'll have bipolar neurons attached to taste buds. You have bipolar neurons attached to, or that help make up the retina of the eye. You have bipolar neurons that are in the cochlea of the ear. So they're receiving some type of special senses. At least that's the main thing we associate those with. And then the pseudo-unipolar neurons were the ones that are found in what's called the dorsal root ganglion. And all that's in the notes. But the reason they're called multipolar, bipolar, and pseudo-unipolar is just based off of how they look. So the multipolar neurons are the ones that we end up drawing the most and talking about the most. And a very simple version of that could just look like this, like a little star pattern, where you have these shorter processes coming off, and those end up giving little branches called dendrites. And that's actually called dendritic branches right there. And then you'll have a long process, a singular long process, and it will end up forming these things called arborizations. That might be a new word. We seldom use it, but at the end it branches again and forms these what's called axon terminals, or terminal buttons at the end. And in the axon terminals, that's where we store the neurotransmitter. So if we're able to send an electrical signal down the axon at the appropriate, say, voltage, that can trigger the neurotransmitters to be released, and that'll cross some type of gap or synapse, or a myoneural junction, whatever you want to call it, depending on what it's attached to. And that's what's going to communicate with the next cell in the sequence. So maybe this neuron wants to talk to other neurons. Maybe this neuron's trying to talk to muscle, whatever. But that's how that communication would work. So when you end up having multipolar neurons, you have multiple short processes, a single long process. So there's multiple processes coming off the central soma. And in this example, that's where the term dendrite gets used best, right? So dendrites, we don't really think about dendrites when we talk about bipolar neurons. We don't really talk about dendrites so much when we talk about pseudo-unipolar neurons. But dendrites works most appropriately when you talk about multipolar neurons, okay? So by definition, they're kind of the short processes that carry information toward the soma in a multipolar neuron. So again, that gets talked about very generically. And if they ever ask you what direction of flow does a dendrite carry, well, it always carries information toward the soma or toward the cell body, right? But axons can carry information toward a soma. Axons can carry information toward a cell body, but they're much longer. So dendrites, what are they? They're short processes that carry information toward the soma or toward the cell body, typically of a multipolar neuron, right? So when I draw this, you'll end up seeing me draw something that probably looks more like that, sorry. So mine will look a little bit more like that. It's beautiful. There we go. That's a nice one. When you get into bipolar neurons, the reason it's called bipolar is because you'll usually have some type of central soma that looks something like this. So just imagine this is the cell membrane, or the neurolemma is a fancy word for saying a neuron cell membrane. And then you'll have two processes that come off that. So that's where the term bipolar comes from. So imagine this is one of the poles of the soma. That's another pole of the soma. So it's a bipolar neuron. And it just really depends, what it's attached to for the direction of flow. Like if CNS is here and this is the place where we're receiving the information, then the flow will go that direction. So you're always heading kind of towards CNS. So that's why these are nice when you get into special senses, because when you think about vision, it's always going in one direction. We're always carrying information from our retina to our optic nerve to the occipital lobe. When I think about taste, it's always going from the taste buds toward the brain. When I think about auditory sense, it's always going from the cochlea toward the brain. So bipolar neurons are good for that type of information. When you get into pseudo-unipolar neurons, these are the weird ones because there's really only one place we talk about them. So they end up looking something like this. And it appears as though they only have a single process coming off a central soma. But in reality, if you got much closer, you would see that that's actually two processes. So if we kind of zoomed in on that little part right there, it would actually look more like this. So technically, sorry about that. It's hard to get everything on here. But it's technically a bipolar neuron, but the two processes have kind of merged together. So we just end up calling a pseudo unipolar neuron. And the thing we want to kind of know about this is, well, where do you find it? And that's typically the dorsal root ganglion. So with multipolar neurons, think CNS primarily. With bipolar neurons, think areas of special senses. And with pseudo unipolar neurons, you end up thinking about what's called the dorsal root ganglion. So again, I'll review, but it's not a bad thing to review. When you get into function, function's fairly straightforward. And you can do this with the nervous system as a whole, because sometimes the entire nervous system is subdivided this way. Functional divisions of neurons is either sensory or motor. And sensory tends to always carry things afferently. Motor tends to carry things efferently. This is picking up some type of stimulus. It's receiving some type of stimulus. And a lot of times it's going to have a specialized ending that can pick that stimulus up, called a receptor, right? So maybe it has an ending that picks up temperature, and then it's a thermoreceptor, or maybe it picks up a chemical concentration or change, and then it's a chemoreceptor. So pH would be an example of that. Blood oxygen and blood carbon dioxide saturation is a chemical concentration that we can pick up. So they'll have specialized nerve endings called receptors. And again, they're always going to be carrying information toward CNS. So that's where the word afferent comes in. Afferent's always going toward CNS. While motor is always going to be carrying information away from CNS, because we're going to go out to things called effectors, right? You're going to go toward things called effectors. And effectors were, I'm just giving you key terms here. These don't necessarily have to coincide with each other. Afferent, efferent do, but stimulus and effectors don't necessarily have to coincide with each other. But effectors tend to be either muscles, MM is an abbreviation for muscles, or glands. And depending on what type of muscle you end up going to, can make your motor neurons tend to veer toward maybe a somatic portion of the nervous system versus an autonomic portion of the nervous system. So again, since neurons are the functional units of the nervous system, when you put a lot of these things together, you end up getting into functional divisions of the nervous system. And that was something that we had seen earlier, where when you get into the PNS, the peripheral nervous system, we're sometimes sending things just to skeletal muscles. And if that's the case, that's where somatic motor neurons would come in. So what type of neurons carry information to skeletal muscles? Somatic motor neurons, right? Because somatic, the word itself means body. But one of the words I told you to think about when you're dealing with somatic in the nervous system is it probably has something to do with consciousness or awareness. So if I'm consciously able to feel something, that might be some type of somatosensory thing. But if it's somatomotor or somatic motor, I'm going to something I can consciously control. And there's really only one thing we consciously control, and that's skeletal muscles. So skeletal muscles would receive signals from somatic motor neurons. So it's just a lot of terminology. Again, hopefully most of it's familiar terminology. And I will recap this with a very brief overview. But this is why they like to go in and classify everything in the nervous system, because you could just kind of keep subclassifying, and you could keep subclassifying, and you could keep subclassifying all day long. But when you get into the autonomics, these are going to go to things we don't consciously control. So if I want to send a signal to the heart, a heart's an effector, certainly. So the signal is motor, but it's going to be more coming along sympathetics or parasympathetics, right? So that's autonomic in nature. So pretty much anything other than skeletal muscles that we're going to try to control organ-wise. So maybe the walls of vessels, the walls of digestive organs, anything you don't have to think about is controlled, through autonomics. And that's another subdivision of the motor portion of the nervous system. So simple version, neurons, which are the functional units of the nervous system, they're doing all the work of the nervous system that we associate with as its key roles, which is, again, just responding to stimulus. So because there's a lot of different types of neurons, we subclassify them, and the two biggest subclassifications of neurons is shape and function. So function is either sensory or motor. Shape was multipolar, bipolar, or pseudo-unipolar. And then sometimes they'll ask you to also kind of memorize, well, where do I find this particular shape of neuron? So if we go back to our little PowerPoint here, we can see, I did put those in for you again. So multipolar neurons, multiple processes coming off the central soma, think CNS, bipolar neurons, two processes coming off the central soma, think special senses, and so on and so forth. So we got that all covered. If you hear a lot of snoring and crying in the background, it's not my wife, it's Oscar, my dog. So he's suffering through this with us. And again, you got sensory neurons, interneurons, and motor neurons. The interneurons we didn't mention just yet, but they're usually shorter neurons. That sometimes gets brought up. I think the only time, and I did put this in your notes for you, that you might be asked about an interneuron. The role is pretty straightforward. They connect the sensory and the motor portions of the nervous system. You could also think about these as being processing centers, because when I'm sending sensory information toward an interneuron, it's got to kind of decide, well, where does this go next? There's like a decision to be made. So they're processing portions, but they're also short usually. So sometimes they'll ask about which of the following types of neurons are unmyelinated or have unmyelinated axons. And it's usually interneurons because they're short. So sometimes a picture, a very oversimplified picture of an interneuron might look something like this, where you have some type of sensory neuron signal coming in. And then you'll have this little short thing, and then they'll draw this, and then you'll see another little circle, and then you'll see another little loop coming back out. And sometimes that's called a reflex loop in a way, where you have sensory information coming in and interneuron connecting, and then you have motor information going out. But they'll tend to draw this little short little object in between. That's your interneuron. So if you're traveling over long distances, like this would appear to be, or this would appear to be, these things typically are myelinated, because if I myelinate an axon, then you can do what's called saltatory conduction. I can just jump from one node of RANVA to the next. So it speeds it up and it maintains the amplitude or the intensity of the electrical signal. Same thing here. If I want to travel over a long distance, I want to myelinate that because I want to make sure it gets there quickly. We don't want to pause and wait. If there's a giant sharp object coming from my eye, I don't want to think about it like this and say, oh, I should probably move. It's got to be quick. But since this is going over such a short distance, oftentimes interneurons are unmyelinated. So that might end up being like a test question for you. Maybe not on my test, but it might pop up one day. They sometimes ask, which of the following types of neurons are unmyelinated interneurons because of their length? So I think I did put that somewhere in the PowerPoint for you, but I'm not specifically sure where. Let's see. This is an example of a reflex loop. It's pretty small. You would have to zoom in on probably your own screen to be able to see this, but they're kind of showing some type of sensory afferent signal coming into the cord, entering the dorsal horn here. And inside that, they're showing a small interneuron, and then the motor efferent signals coming back out and going to a muscle. So this is typically the example I give you in class. Glial cells, not a lot to say here. Make sure you memorize them. They're easy to ask about. Which ones clean up spaces, which ones form the blood-brain barrier. That certainly might come up with your pharmacology class, because sometimes you have to design drugs that can bypass the blood-brain barrier. Which ones form myelin? And, if you're forming myelin, they will be different. Like if I'm forming myelin in the PNS, it's Schwann cells, but if I'm forming myelin in the CNS, it was oligodendrocytes. So again, we've seen that before. Just make sure you know which glial cells do what, but there's no need to discuss them too deeply. Myelinization, we already mentioned this concept as well. Myelinization is a process of applying a fatty sheath. Big picture wise, it's what produces white matter in the nervous system. So if I've failed to emphasize that before, white matter is primarily myelinated axons. It shows you where information is being transmitted. But why do we use myelin on axons? It's, again, to speed up the conduction. Typically, the more myelinated it is, the faster it can travel, and it also maintains the amplitude. So I did put that for you down here, because ultimately, when you end up going through that depolarization phenomenon, we want to make sure that the voltage that we generated stays as close to its original voltage as possible by the time it makes it to the end of the axon. So that's what they mean by maintaining proper amplitude. Because ultimately you're going to need a certain amount of electricity to cause the neurotransmitter to go through exocytosis. So if we don't have properly myelinated axons, then you might start off with a high spike on your graph, but by the time you get to the end of the axon, it's diminished and you don't have enough anymore. So the signal might not transmit. So we'll talk about problems with that in just a second. This talks about the type of conduction again, saltatory conduction. This is a good analogy. I've mentioned this before back in our Anatomy 1 class. So if you didn't have me, just kind of read that. It's a good way to think about how myelinated axons work versus unmyelinated axons. I never make people memorize these lists when it talks about nerve conduction velocity, but it is a good point we can make. Certain things are going to travel much faster along myelinated axons than others, and sometimes it has to deal with, well, how myelinated is it? Because if I end up having more myelin, it's going to travel faster. If I have less myelin, it travels slower. So that's an important concept. I'll mention an application of that in just a minute, but if you start demyelinating somebody, maybe the symptoms won't be as bad at first, but as the disease progresses and you have less and less myelin, you can certainly maybe have some type of direct correlation. Well, I'm going to have less conduction too. So the signal's eventually going to get lost. The other application is if you've ever had pain before and you went and you pressed on it, or if you had maybe like an itch and you went and you pressed on it, you didn't notice it as much anymore. It's because you can sometimes bypass pain and itch with mechanical compression. So if you've ever had like an ache before and you pressed on it and all of a sudden the ache went away, it's because you pain gated it. Sometimes when neurons send signals, they sometimes have to share a route. And so let's say that we have a singular route, and this isn't entirely accurate, but it's close enough. Let's say there's a singular route I need to pass through to get to my destination somewhere up here. And you have this heavily myelinated axon that's coming in here, and then you have this slightly less myelinated axon here. This signal is going to travel much faster than this one does. So sometimes pain is less myelinated than, say, mechanical compression or touch. And so if pain's traveling at this rate or speed and mechanical compression or touch is traveling at that rate or speed, then by me pressing on the area that hurts, that signal is going to get there faster and it's going to be able to go through the gate more easily. This one's going to end up being blocked and it's going to have to wait for the traffic to diminish. So it's an interesting concept, but again, nothing we need to dive too deeply into. And it's the same reason TENS units work. So if we understand the basic point of myelin, what would happen if something becomes demyelinated? The simple answer is the signal will not get there as well. So this is what we end up seeing with certain diseases like, say, multiple sclerosis or possibly Guillain-Barré, where you have certain demyelinization that's occurring. Exact causes are hard to guess. Sometimes they'll throw in some type of genetic component. Sometimes they're going to say it comes after certain viral infections. Sometimes it's going to, I'm sure we'll find out one day, but at the end of the day, when you have a demyelinating disease, the signal just doesn't travel as well. So with multiple sclerosis, one of the most famous demyelinating diseases, you end up getting one of two types. The more common type of MS is what was called the relapsing remitting type, where you would have symptoms that would constantly come and go. But maybe pain would be a symptom, maybe blurry vision would be a symptom, maybe you would have slurring of speech as a symptom, and then all of a sudden it clears up and it's gone for months at a time, and then it comes back. So I had a friend one time that had a similar condition called Devick's disease, and hers would only show up in the summertime. And that's not completely unheard of, because if you look at the bottom of the screen here, there is something called Uthoff's phenomenon, where symptoms sometimes get worse with heat. So another thing that they've sometimes noted about multiple sclerosis is this tends to be hidden better when you live up north. But if you end up traveling from up north to down south, and it starts to, you have that climate change, you start to get into hotter environments, it tends to exacerbate the condition, or vice versa. If you end up going into colder environments, the condition seems to relapse a little bit more. But regardless, when you have these demyelinating diseases, whether they're relapsing, remitting, which again, that helps with the diagnosis, because if somebody says, I have all these conditions, and then they go away for a short amount of time, and then they come back, and then they go away, that helps you with the diagnosis. But many, regardless of how they start, will oftentimes become, or at least seem to become, progressive. Because even with the relapsing remitting, sometimes the distance in between, or the time in between the symptoms showing up, gets shorter and shorter. So as the disease progresses, it's going to relapse more quickly. So either way, it'll kind of end up being progressive in a way. More common in females, usually around the 30s is when they'll start showing up. Charcot's triad is made up of 3 early signs and symptoms. So this might be some of the early things they notice. Might not be how they say it, but it could be something you could pick up on objectively. So again, just for your information, a sign is something that the provider or observer sees. A symptom is something that the patient describes. And scanning speech might be noticeable early on. Scanning speech is where they tend to have these breaks. And sometimes it just seems like they have a hard time getting it out, or maybe they'll emphasize things at weird points. So scanning speech, I think it's sometimes called staccato speech. Don't quote me on that one. You can look that one up on your own. They sometimes end up having these weird emphasis on different syllables that most of the time you wouldn't. So that's sometimes called scanning speech. Might appear like a drunken speech as well. Intention tremors is when they tend to be shaky when they're trying to grasp an object or move toward an object, like say with their hand, something like that. It's also kind of a sign of dysmetria, and I'll mention why that's important. Hopefully you can kind of see that like in my screen up top. I know the screen's not really working too good there, but if I am doing something with intention, you can tell what I'm trying to do here, but it just maybe doesn't look right. And I think my camera is actually clearing up some of the shaking that I'm doing there. But that's intention tremor, when it looks like somebody's trembling when they're trying to do something. We oftentimes associate that with cerebellar dysfunction, right? Dysmetria also looks like that. Like dysmetria sometimes looks like you're clumsy because you have trouble judging distance between objects, so your hand-eye coordination is off. But the reason that a cerebellar problem and MS tend to overlap is because that's where MS is most frequently going to kind of show up first, upper cord or cerebellum. So that is common amongst cerebellar tumors or patients with MS. And if you have lesions caused by demyelinization, then it would appear the same. And then nystagmus is something that you might notice, but the patient wouldn't describe it that way. The patient can't see that they have nystagmus, so they might say that they have bouts of blurry vision. Like they have blurry vision and then it becomes normal, it goes away, and then all of a sudden they got blurry vision. So nystagmus is the shaking, though, of the eyes. So again, we see that with cerebellar dysfunction. Let's see. So here's that. Symptoms come and go. That's, again, it's helping with the diagnosis, relapsing, remitting. Lhermitte sign, this is a way we can test it. Sometimes you could call this a neurological test. Sometimes you could call this an orthopedic test because you are having them kind of assume a certain position. But if you have the patient flex their chin to their chest, or maybe you flex their chin to their chest if they're supine or laying down, if they notice that there's electric shock-like pain upon neck flexion, that's a good indication that they do have MS. Let's see. So no cure. So most of the treatment is symptomatic. A lot of times when you're dealing with inflammatory conditions, you'll probably notice that steroids would tend to cause these things to feel better. So anytime you have inflammation, autoimmunity, a lot of times steroids ends up being part of the treatment plan. Axon injury, kind of a duh idea here. If axons are the things that are transmitting the electrical signal, if the axon has been injured, then clearly the electrical signal can't get there. And that's obviously true. So if we take it one step further and think about, well, how damaged is the axon? You know, is this a concussion where I'm going to expect everything to kind of go back toward normal, or at least hopefully go back toward normal? Is this some type of burner or stinger where you have some type of traction injury? Is this actually been completely cleaved? Has the axon been completely torn and separated? And if so, how much? Those all play factors, but at the end of the day, if the axon has too much damage or forms too many fibrotic lesions on it, then the signal will no longer be able to be sent. So at that point, we might want to determine, well, where is this problem occurring? Is it occurring in the CNS or is it occurring in the PNS? Because one kind of has a little bit of hope of being fixed and the other one tends to not. So typically, if you get axon injury in the CNS over time, it's going to, again, form lesions, maybe scar tissue in which signals are no longer able to transmit, and you would lose functionality. In the PNS, there tends to be a little bit more hope of possible, I guess, not necessarily completely cured, but progress and improvement. And again, this would depend on to what degree was an axon injured and how much. Because if there's too much of an injury, your axon will tend to wither, or it goes through what's called Wallerian degeneration, and you just end up losing function as if the axon were no longer functional at all. But if the axon injury is kind of minimal, sometimes it'll regrow and it'll almost seem as there was no injury at all. This is a basic concept. I don't want to spend too much time on this one. Nerves and neuron are not the same. So even though I make the exact same mistake, I'll sometimes say nerve when I mean to say neuron. A nerve's an organ. It's something we can see with the naked eye. It's made up of thousands of axons, and those axons are surrounded by a sheath. And then there's another sheath, and so on and so forth. So it kind of looks like this. So every little individual circle you're kind of seeing is where an axon is being surrounded by something called an endoneurium. So there's thousands of those things, and this would be the overall nerve, like maybe like a median nerve or something like that. But at the end of the day, it's in very simple terms. Like let's say I were doing like some type of motor nerve. Let's say you have a cell body here, a cell body here, a cell body here, axon, axon, axon. Wrap these up in a couple of sheaths, and now you have a nerve. That's probably the simplest rendition of a nerve I can give you. But Here's a neuron, cell body, cell body, cell body, axon, 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 nerve. Okay, it's just axons wrapped up in a sheath. Sometimes there's multiple directions. If that's the case, then it's what's called mixed. Sometimes there's a single direction. And depending on which direction it's going, will determine whether it's sensory or motor. So if I'm going towards CNS, then it's sensory. If I'm going toward effectors, like out in the body toward muscles or glands, then it's motor. If I'm having signals go in both directions, this way and that way, then it's what's called mixed. And when you get into the nerves, that's the ones that we sometimes will number. Like in the CNS, we have 12 pairs of cranial nerves. In the PNS, we have 31 pairs of spinal nerves. And then the nerve fibers, again, this is where some of that early terminology came from. So there's no harm in going over this again. When you get into nerve fibers, let's just pretend we have a sheath and we're not going to look at the origin. We're just going to see some fibers. Let's say this is the outer sheath. Let's say this is the sheath right here. I'm just going to make this a little thicker so it stands out. And let's say we have a fiber here, a fiber here, a fiber here. Let's say the body is out here. Let's say the CNS is this direction. If we're going in this direction, that's afferent. If we're going in this direction, that's efferent, right? Efferent, if you want to remember, exit CNS. So we have CNS right here, and we're going in this direction. We're exiting the CNS. That's an efferent signal. Efferent is always going to be motor, right? So efferent signals are always motor. Afferent signals are always going to be sensory. So I'm sorry you can't see that, but I'm going to go on space here. Efferent exit. There you go. So when you get into somatic, think about things that you are aware of or you can consciously feel or control. If it's visceral, that goes better with autonomics. It's stuff that's kind of taking place in the background. We are not aware of it. This is controlled autonomously. And it's sensed autonomously. Like you don't even have to put thought or effort toward that. So sometimes, again, I would be, it's somatic. There's conscious awareness or conscious control. If it's visceral, it's subconscious, right? That's another way to think about it. So to determine whether something is traveling along a somatic motor or visceral motor, somatic afferent, visceral afferent, you just have to ask yourself a couple of questions. So let's look at the terminology real quick again. If you somatic afferent, just break the word down. Afferent is going toward CNS. That means it's sensory in nature. What are things that I can consciously pick up sensory wise? Well, maybe touch, maybe itch, burn, pain. Those are things I'm all aware of. Proprioception is something I can pick up that's somatic afferent in nature. So sometimes it's just, if it asks you, you know, which type of signal or which nerve fiber would carry touch, just ask yourself, well, what is touch? Is touch something that I can feel, or is touch something that I'm sending to a muscle or a neuron to do, right? So if I'm picking up the sensation of touch, if I'm picking up the sensation of touch, that's something that I feel, that's sensory. Am I consciously aware of it? Yes. Let's say itch. That way we don't have an action, you know, associated with it as well. You know, let's do pain, because sometimes I say I'm itching a bug bite, so let's not say itch. But let's say pain. Pain is something that I feel, right? And that is going to be afferent in nature. I'm also consciously aware of it, so that would be another type of information that travels along a somatic afferent fiber, okay? Let's do visceral afferent real fast. So again, let's go in the same direction. We're going to feel something, but I'm not consciously aware of it. Maybe my brain is somewhere in some type of nucleus inside my brainstem or in my hypothalamus, but it needs to monitor it. So we're trying to pick up stimulus from the body. So if it's visceral afferent, that's sensory information of something that I'm not normally consciously aware of. So maybe the composition of food in my small intestines. That's something we monitor. Maybe it's the pH. Maybe it's the pH of the blood. Maybe it's the oxygen concentration of the blood. Maybe it's the carbon dioxide concentration of the blood, right? So those would be more visceral in nature. Afferent is just telling me we're sending sensory information to be processed. If we're doing somatic efferent, that means we're going out. So we're going toward effectors. Effectors tend to be muscles or glands. So somatic efferent, there's one thing, it's skeletal muscles. If you're doing visceral efferent, that's a lot of stuff. Anytime I'm controlling smooth muscle, cardiac muscle, so the heart is going to use visceral efferent signals to speed it up or slow it down. The digestive system is going to receive visceral efferent fibers to speed it up or slow it down. So a lot of times the easiest ones to remember are the somatic afferent, the somatic efferent, and if it's not part of that group, it's visceral afferent or visceral efferent. So again, somatic afferent things are things that we can consciously feel, if you want to think about it that way. So that would be stuff of the skin or joints. Somatic efferent are things we can consciously control, so skeletal muscles. Visceral afferent is coming from like the organs, the vessels. Visceral efferent controlling the things that I don't have to consciously think about to control it, like blood vessel, vasoconstriction or vasodilation, peristalsis in the digestive system. So again, that's kind of a deep review, but fairly inclusive of any type of question that could come from it. This reason this kind of sits in this one position, peripheral neuropathy, typically a disease that at a very small level. Usually when you're trying to supply nerves with blood, that actually travels in little teeny tiny vessels. So the very smaller vessels. And those will go on along the endoneurium. Sometimes you'll see some of the vessels embedded in certain cross-section diagrams where they'll show color. But they're very small vessels that are in here to help supply the axons and keep the axons alive. And when you have diabetes, the long-term effects of diabetes is damage to the smaller vessels. The smaller vessels tend to die. So if I can't keep the vessels alive, which are supplying these very small axons, then we're certainly going to see damage and degeneration of the axons as well. So when you have a patient with long-term diabetes, we tend to see the death of those axons occurring distally first. So this is where diabetic peripheral neuropathy comes from. So think about the tips of the fingers, the bottoms of the feet, the tips of the toes. That's where we tend to see diabetic peripheral neuropathy start, and it's usually due to the death of the small vessels that are supplying those little tiny axons in the nerves. So maybe they'll notice paresthesia at first, the numbness and tingling. It can turn into causalgia, which could turn into pain, but eventually it's going to turn to weakness and total numbness. Like numbness and tingling is usually an early sign of neurodegeneration. Just numbness, though, that's a later sign. That's usually it's dead, right? So numbness and tingling early. Just numbness with no tingling left, not so good. Starts unilateral, becomes bilateral. This is actually a good sign that this is something greater than Oscar is snoring in the background, if you can hear that. This indicates it's something bigger than just a small impingement, or maybe I slept on my arm too long, or maybe I hit the back of my leg and that's causing my foot to become numb. When things start to become bilateral, that tends to imply this is either a more centralized in the CNS or this is a more systemic thing in the body. So I like it when patients used to come in and they only had, unilateral pain in, say, the pinky finger. because that told me it probably didn't start as a problem until you got at least to the wrist on one side. It helped me localize that lesion. But if both of their hands were numb, I was concerned, right? Because now it's gone bilateral, so that means it's more cord or brain oriented, possibly. So that's just it's a worse sign. So if you start to get a lot of numbness and tingling, causalgia in both hands and both feet, again, that indicates there's a bigger problem going on. Obviously, if I can't feel the bottom of my feet, think about that. I'm going to have difficulty with balance and walking. And a lot of the treatment tends to go toward that at first. So you might have heard of what's called proprioceptive training, where you're going to try to reteach them how to balance the body when they can't even feel their feet. So maybe they start using those. They have wobble boards. They have these little inflatable. It's almost a stability ball, but it's like an inflatable pad that they stand on, and you have them do different types of balance exercises. That's usually called proprioceptive training. Different types of orthotics for the feet, making it more comfortable, all that kind of stuff. But again, this is only going to become progressive if the diabetes is not adequately hit. So this is certainly not something you're probably unfamiliar with now, and you're going to become a lot more familiar with it later. But if you're looking at this from a big picture, this is more of a nuisance. The bigger idea is we have poor blood circulation going to the extremities, and it's not only causing the nerves to die, but what happens if you get an infection, right? So if I don't have good circulation going to the nerves, there's chances are not good circulation going to anything. So if a patient ends up getting an ulcer in the bottom of the foot, it's only going to fester and get worse. So it's becoming the bigger deal. But this is a good indicator that it's happening. Let's see. Give me 1 second here. So when you get into spinal peripheral nerves, this is something that we weren't as focused on this part of it as we were this part. So let me draw this picture for you real quick, so it's a fresh reminder. One of the things that we emphasized back in Anatomy 1 was the direction of flow when we're trying to bring things from, like, say, the body to the CNS and the CNS back out to the body. And the first place we started talking about that was in the cord. So we ended up drawing a cord cross section that looks something like this. And mine never really all that symmetrical, but you end up having the outer portion, which is, think about that as like the white matter here. And then you have this inner portion that's darker. And so this is usually referred to as the gray matter, like all of this. All that's gray matter. All of this is white matter. So wherever you have gray matter, that just means you usually have a large collection of the cell bodies. You know, wherever you have white matter, it's large collections of axons because the axons are myelinated. So just think about having a cell body here and a cell here and a cell body here and a cell body there. And maybe some of their signals are going to end up going up this way. Like if it's going to dorsal columns, maybe some of their signals are going to come down here if they're acting as an interneuron between the dorsal horn and the ventral horn. So, but gray matter versus white matter. Out here, we tend to think about tracts. Again, in here, we tend to think about cell bodies. And that quick description, let's go one step further. Let me get rid of that so we can see it through the paper. But if we have our spinal cord cross section, I'm going to tend to emphasize this side over here. I'm not going to draw everything bilaterally. But let's say we have this dorsal horn right here, and we have this ventral horn right here. Dorsal horn is always going to be receiving some type of information. So the information was coming from the body. It got up to the dorsal root ganglion, communicated with a cell body right there. This said, all right, that information seems important. Let's go ahead and send that to the dorsal horn. And its communication would happen somewhere in here, or it might bypass it and go somewhere a little bit higher. But either way, it's going to enter into the dorsal horn. And one of the simple concepts we talked about when we were doing spinal cord cross section. How do we link everything up together? You had a sensory afferent signal coming in, that information was going this direction. And then I ended up showing you the motor efferent information coming back out again. And that was heading back in this direction. So this was motor efferent. So down in this ventral horn, we have these things called alpha motor neurons. And alpha motor neurons are things that go to skeletal muscles. So that was probably talked about in maybe a separate chapter. Maybe that was talked about more in the muscle chapter, chapter 9. But regardless, this is receiving information in the dorsal horn. This is sending information out in the ventral horn. And if you were to take, say, lots of these signals, and then wrap them all up in a sheath, now you have a spinal nerve, right? And we didn't really get to this whole concept. We try to keep things in little separate bundles. But there's also another contributor that can go along in this nerve, and sometimes it's just going to travel in a small portion of it. But there's also what's called a lateral horn right here, which can send out autonomic information. So let's not draw that, because you end up having to form these little loop-like signals. So if we look at a typical regular peripheral spinal nerve, so this is going to be a peripheral nerve. It's obviously attached to the spinal cord, so this is going to be a peripheral spinal nerve. You can already see how these would all be mixed in nature, because mixed means I'm carrying both sensory information and motor information. So maybe something comes in, like the sensation of touch, itch, burn, pain, and then maybe a signal go out as, all right, skeletal muscle contract here, right? Obviously, the location of wherever we are in the spinal cord would determine which muscles we're going to, because they're specific. They follow things called myotomes. But regardless, sensory information is coming in along this thing, motor information is going out along that thing. So all spinal nerves are built this way, right? So peripheral spinal nerves are all mixed in nature, because they're both sensory information and motor information. Some information is going toward CNS, some information is going away from CNS. So that's all peripheral spinal nerves. And then I'm going to skip over this for a second. Eventually, let me see where it is in the notes. I want to do numbers with you real quick, because while we're talking about it, I think we got something here that's going to talk about that. You end up seeing 31 pairs of those. So I ended up planning asking you about how many comes from each section, but let me just go ahead and explain it now while we have this. So when you get into the peripheral spinal nerves, there's 31 pairs total. And I'm trying to find the slide for you that has all that information. But I kind of want to talk a little bit about just this real briefly. And I don't want to waste too much time looking for this, because I know it's in there. Here we go. So 31 pairs of spinal total, 8 cervical, 12 thoracic, 5 lower, 5 cervical, and 1 coccygeal pair. Let me show you why it is. In most parts of the spinal cord, we are exiting down, and I'm going to maybe draw this from different points of view. So imagine this is a spinal cord. Here's like a dorsal root. Here's a ventral root. Here's a peripheral nerve. Here's a dorsal root. Here's a ventral root. Here's a peripheral nerve. You know, how does that relate to the spinal cord cross section? Well, if we have this right here, if we have the little spinal cord section, here's the dorsal horn. Here's the ventral horn. Let me move that up a little bit better. There we go. Dorsal horn, ventral horn. This part right here is called the dorsal root. This part right here is called the ventral root. Think about the spinal nerve like where it begins as the trunk. So you have a root, a trunk, right? And the dorsal root ganglion is just a collection of cell bodies on that dorsal root. So you got a root, a trunk, right? So that's kind of what I'm showing here. root, you got the trunk, so dorsal central root, trunk, dorsal root, trunk. That's a spinal nerve, that's a spinal nerve. Normally, the way these things are numbered is based off the vertebra that sits above it. So if I'm in the thoracic region, if I'm in the lumbar region, that's going to be the case. So when you look at, say, like the T1 vertebra, exiting just beneath that between T1 and T2, you end up having the T1 spinal nerve. So it's numbered accordingly. But it doesn't work with the cervical region. So when you get up in the cervical region, imagine we have our C1 vertebra, C2, C3, C4, C5, C6, C7, right? There's 7 cervical vertebrae. 1, 2, 3, 4, 5, 6, 7. I just told you in the thoracic and the lumbar region were named based off the vertebra that's in the cervical region of the neck. We have an extra spinal nerve that goes out above C1. So now that makes this one C2, C3, C4, C5, C6, C7, C8 spinal nerve. And then you start doing the normal number scheme. So we end up having an extra cervical spinal nerve in the cervical region because you start off with one that goes above the first cervical vertebra. So that ends up being C1. That's C2, C3, C4, C5, C6, C7, C8. So I figured I'd just show that to you while we were on it. And then once you get to the thoracic region, now you have the T1 spinal nerve. This is the thoracic region. This is the first one. This is the second thoracic vertebra. So there's the T2 spinal nerve. Okay. So then it kind of follows suit with the normal numbers. But maybe that clears it up, because I know when we're in osteology, we're like, there's 8 cervical vertebra, there's 12 thoracic, there's 5 lumbar vertebra. But then when you got spinal nerves, there was always that one extra. That's why. So 31 pairs of spinal nerves, all of them are mixed in nature. They carry both sensory and motor information. Read most of this. It just kind of talks about, where the spinal cord ends. The conus medullaris is a visible structure. Like if you were ever present for what's called a laminectomy and you actually see the back of the vertebra removed, there's a procedure. Let me see if I can get rid of this. Imagine you have a vertebra that I'm looking at from the side. This is your vertebral body. So let's say this is in the front, this is in the back. You end up having pebbles on sides. You end up having a lamina where they meet, and then you have a spinous process, and there's a transverse process as well. So this is just kind of a view from the side. When you end up doing what's called a laminectomy, you cut off the back portion of the vertebra, and it exposes the cord that's sitting right here. So sometimes that's done to take pressure off the cord. If I'm looking at, like if we were to take this picture and flip it that way, when we look inside, eventually you'll notice that the cord comes to a point and it tapers like this. So the reason I've seen this before is we used to do laminectomies on the cadavers in the lab. So so eventually you will end up seeing this structure, and I can show you a picture on the internet in a minute. So if we've done a laminectomy and we put the back, you'll see the cord eventually gets down to a point, and that point is called the conus medullaris because it has this cone shape and it's right in the dead center. So it's a little cone shape in the middle, conus medullaris. And then you'll end up seeing a little dangly, thicker cord coming off that, and that's called phylum terminale. And this will eventually attach all the way down to the coccyx, and it kind of forms an anchor point for the cord. But once you get down to L2, L3 space, that conus medullaris typically forms. So let's say that's L1, here's L2, here's L3 vertebra. This is where conus medullaris typically forms. Here we still have a spinal nerve coming off. Here we have a spinal nerve coming off. Here it's going to change a little bit. We're going to have a spinal nerve coming off here, But then we still have other ones that are going to come off here and here and here and here. So when they exit together like this, they end up forming this little bundle at the end, and that's called cauda equina, right? So obviously the cord is already terminated, but we're still sending off spinal nerves, which will exit underneath vertebra because there's little spaces in between adjoining vertebra. Like again, if I go back to a side view of a vertebra, you'll have this little space right there where the spinal nerve can exit. Right? And it's formed largely by some of the various different structures that make up the vertebra, but it's a little safe space where it can exit. If it gets occluded, then you're going to get nerve kind of pinched in that occlusion. But once you reach the end of the cord, we still have nerves that have yet to exit. So they come off in a bundle. So that bundle is called cauda equina. So a lot of times I showed you that in a picture, but this is how it forms in that nature because the cord itself has already terminated, but we still have more spinal nerves that have yet to exit. So they kind of come off in a group or a bundle. So I think we have a good side view of that here. In a way, you can kind of see that. But there is something specifically that's called cauda equina syndrome. And that is typically noted when a patient has lost all bowel and bladder function. They can't feel the inner thighs. So they sometimes call it what's called saddle paresthesia. So just imagine anything that you could possibly come in contact with if you're sitting on a horse saddle, anything that comes in contact with that saddle, you can't feel. That's called saddle paresthesia. So if a patient ends up getting a bad disc rupture or maybe like a bad low back injury, what can sometimes happen is the material. Oh, I'm sorry, I forgot to share with you the video here. So what I was pointing at was when you sometimes get what's called cauda equina syndrome, what happens is a disc has ruptured and some of the material that was inside of the disc it's caustic. It can cause a burn type sensation that ends up squirting on those spinal nerves, and it causes them to have temporary paralysis, if you will. So cauda equina syndrome it's a big problem, but again, it would probably come from some type of really bad compression injury. So what I was going to show you was, imagine you're looking at the low back, and you can see these on MRIs. Imagine you have a vertebra right here, and then there's another vertebra right here, and then there's another vertebra right here, and then there's another vertebra right here. In the low back, you tend to get what's called a lordotic curve like this. It gives you that kind of roundedness of your low back. So your discs tend to be pie shaped in this direction. So your discs are kind of more bulged toward the front, a little bit smaller and flatter in the back. So sometimes if I get too much axial compression, like too much pressure going this direction, what will end up happening is it can cause this to compress on that, for example, and that'll take the material. And if there's enough pressure, that material can actually prolapse through the hard, what's called annular fibers of a disc. So if that's a vertebral body, you have a disc, vertebral body disc, vertebral body disc. So inside of a disc, there's something called a nucleus pulposus, and it's more of a jello-like material that can protrude out. And if it ends up squirting some of these spinal nerves, especially down in the lower back area, that can lead to what's called cauda equina syndrome. So that's, again, usually some type of hard compression injury that would cause that. Let's see. I'm going to take a break here, so I'm just going to pause it. You're not going to notice anything, I'm sure. You need a break as well, but I'll be back in a few minutes to pick this recording up. So picking up right where we left off, here's an image of what's called saddle paresthesia. This would be the area that a patient might note numbness and tingling. So again, imagine you fell off a bike and you landed on your butt and maybe you peed yourself a little bit, but now all of a sudden it's getting progressively worse and worse and worse. And I don't feel like my reflexes in that area are as good as they used to be. So maybe I can't prevent a bowel movement. And then progressively, as it gets a little bit worse and worse and worse, you might notice that you can't feel that area at all. So in this area, again, we would probably be inclined to think that cauda-equina syndrome had taken place. So here you can actually see on one of these images, let me make this a little bit larger, the material that was inside the disc here, which is kind of appearing more whitish in color. Here you can see that the liquid material is white. The cord here is dark gray. So here, this is fluid like cerebrospinal fluid. Here you can see the fluid inside the disc. And then all of a sudden you get down here and it's just kind of like a flat wafer. So something has caused a protrusion to come outward, and you can see where the pinching is occurring of these cauda-equina nerves. So again, numbness and tingling in a saddle-like area, or what's known as saddle paresthesia, loss of bowel and bladder control. That is sometimes referred to as cauda-equina syndrome because of the formation that those nerves take toward the terminal end of the spinal cord. Let's see. Let me go back up to something we had skipped over. And I know there's a lot of brain slides we have not gotten to yet, but I think if you remember, the brain, we study that in a fairly simplistic manner. This part is responsible for this, and this part's responsible for that. We're going to try to keep it largely the same. But going back to the earlier discussion we were just having on spinal peripheral nerves, Guillain-Barré tends to be a disease that would be similar in pathology to MS, but the location of what's being attacked is slightly different. So again, when we think about demyelinating diseases or myelin sheaths not functioning the way they're supposed to, imagine signals get lost. And if the signal can't quite get there or doesn't get there with enough amplitude, then we're not going to get the effects that we're looking for. So with Guillain-Barré, rather than taking place in the cerebellum and the upper cord, we tend to see things along the periphery. So Guillain-Barré, similar phenomenon to MS, in which myelin sheath is attacked, but more restricted to the PNS. You may experience both sensory and motor problems because, again, if you remember, peripheral spinal nerves are all mixed in nature. Due to autoimmunity, sometimes what you may end up seeing is a younger individual. And typically, most of these people are very healthy. A lot of times you'll see it on the news, and it's this very athletic young man comes in, and after 2 weeks, his legs have withered away and become atrophied. And then they eventually find out it was Guillain-Barré syndrome that had done it. But they might end up noting that he had just gotten at the end of a sickness. So maybe he had some type of strep throat, or they sometimes note that he was recovering from an illness and he noticed this rapidly ascending paralysis. So yes, it is due to some type of autoimmunity, but they sometimes noted that it's triggered by some type of infection. But the key words that give away Guillain-Barré is this: rapidly ascending paralysis, bilateral in nature, which would distinguish it from, say, polio. Polio is rapidly ascending paralysis that usually starts off unilaterally. So maybe like one leg. Think Tiny Tim. You know, when you think about, like, say, the Christmas Story and you got the little kid with the single crutch, that's polio. So think about bilateral rapidly ascending paralysis and you have Guillain-Barré. So sometimes these people will get weak that starts acrally. That's a word you might not be super familiar with, but acral means toward the tips, like TIPS. And so maybe it starts at the tips of the fingers, the tips of the toes, and it works its way backwards. But again, this would be very quickly. So the person may lose the ability to walk. And then you might notice very rapidly that muscles start to atrophy because skeletal muscles need nerve energy to live and survive and thrive, and can diminish fairly quickly if the nerve itself is no longer sending what's called innervation. So sometimes you'll see a lot of muscle loss in people with Guillain-Barré. Tests include nerve conduction velocity and blood tests. And obviously, the biggest problem will occur. Things don't always stay accurately. When things tend to progress, they tend to move back toward the core. And if our core ends up affecting muscles that control breathing, then obviously that's going to be fatal. Let's see. Just read over synapses. Again, synapses is where some type of axon terminal ends up meeting with its target cell that it's trying to communicate with. So sometimes the synapse is referred to as the space, but it's actually made up of components. You have a presynaptic end plate, the synaptic cleft, and a postsynaptic end plate. And to be familiar with those things, but sometimes with certain diseases, like say myasthenia gravis, it might be a problem up here, or it might be a problem down here, like with the receptor site itself, or when you get into certain types of medications, when you talk about things like reuptake inhibitors, the effect is in here. So it's just good to be familiar with these terms, but nothing really new for us to describe. Here, myasthenia gravis, this is that I just mentioned. Myasthenia gravis, when I think about that word, I tend to think about progressive weakness or slowing down with the ability to use muscles. So this is another, again, autoimmune disease. Hard to tell what's going to initiate it, or again, it may follow some type of bad infection in which the body naturally produced antibodies, and the antibodies all of a sudden see parts of the body itself as looking similar to that disease. It just fought off. So sometimes with these autoimmune diseases, you might have a trigger effect where the person, again, might have had strep throat or might have had a bad strep infection. And then all of a sudden it starts to kick in these unusual diseases. But with myasthenia gravis, I tend to always think about a person's eyes or chewing muscles being affected first. So these don't tend to affect necessarily accurately, where you get the weakness and the slowing down, but they tend to affect muscles you use a lot, consistently and more often. So when I think about eyes are things that you have to constantly keep in focus, and they're moving around a lot. And when you think about your mouth, other than talking, you're chewing and doing things like that. So that's where people tend to start to see the symptoms first. Maybe they would get blurry vision. Maybe they would start to get difficulty and fatigue with the chewing muscles. So maybe that would be noted as diplopia, double vision or hard time keeping your eyes focused. The eyelids themselves might start to droop. So that's called ptosis. I still call it ptosis just to remind myself and remind people there's a P there, but the actual pronunciation of that is ptosis, if you're wondering. This is an interesting phenomenon that they will sometimes use on a standardized test. Gets worse with activity, gets better with rest. So if you're dealing with an issue in which you're starting to get attacks of your synapse or portions of your synapse, sometimes these things get hyperpolarized or fatigued. And when there's damage going on, sometimes if you just relinquish some effects, it tends to clear up a little bit, and then it tends to get a little bit better. So when they're describing that, they're basically saying it gets better with rest, it gets worse with activity. It's just words that they're trying to push you to these diagnoses. So I made sure to highlight it here. Let me go back and see if I did that with Guillain-Barré as well. Here I did put sensory loss and motor problem, but if you want to go back and highlight on your notes or in your PowerPoint, rapidly ascending bilateral paralysis. So these are some of the key terms that will remind you it's one thing versus another. So with Guillain-Barré, you're getting sensory and motor problems, rapidly ascending bilateral paralysis. And one of the key terms when you're dealing with myasthenia gravis is think about you're getting fatigue, loss of function in things that you use often. So again, think about chewing muscles, visual muscles, stuff like that. But they'll oftentimes describe it as gets worse with activity, gets better with rest. Okay. So I'm trying to find as many key terms as I can. Let's see. A lot of times, again, I mentioned this to you before, with autoimmunity, there tends to be inflammatory effects that come with that. Like inflammation is one of the features of an active immune system. So whether I'm trying to clear out damaged tissue, in which phagocytic white blood cells might be used, or whether I'm trying to call in other parts of my immune system to help, certain chemical communicators get released by that. And some of those chemical communicators can trigger part of a thing called a complement system that creates inflammation. So when we do have an overactive immune system, oftentimes inflammation is associated with that. So again, when I think about steroid uses, always think about anti-inflammatory drugs. But again, it's not just going to come from a shoulder issue or a joint issue. Sometimes from our immune system being hyperactive. So anytime you see that, steroids tend to be a common treatment. So with autoimmunity, steroids tend to be a common treatment. With any type of pain associated with joints, overuse injuries, steroids tend to be a treatment because they have very strong anti-inflammatory properties. So finally, getting into the brain. First part, it talks about meninges again. I think this is important to bring back up again. One of the basic concepts is your brain and core are obviously very important. These are the central portion of your nervous system. This is where the processing takes place. It's also easily damaged. It's sensitive to vibrational stresses. It's sensitive to pressure for sure. So there's a limited space between your brain and the skull. There's a limited space between your brain and its surrounding structures of the vertebral column. And filling in that space, have fluid, cerebral spinal fluid, which is going to act as a shock absorber and provide some nutrients. And then you have the meninges. And the meninges kind of serve two purposes. One is it's a physical barrier. It's physically protective. It's holding the brain in place, so it'll keep it from shifting one side to another or front to back. Surrounds, protects the brain. But if any of these meninges, like these protective membrane barriers, get swollen, that's impeding on that little tiny space that we have surrounding the brain and in between, say, the brain and the skull. So that's where that tends to come up most clinically. So what are the meninges? What's the basic function of meninges? And then what are some problems with that we might experience around the meninges? Again, meninges is a protective layer that we use to kind of secure the brain in place within their bony structures. Between the meninges and the brain, we're also going to have some cerebral spinal fluid. But once you combine those things, you're taking about as much space as you have available. So anything that causes swelling, anything that causes something to fill in one of those spaces that's not supposed to be there is going to cause problems. So one of the first things that we probably mentioned back in 101 that's associated with meninges could be a blood clot or a bleed. So we might have mentioned things like a subdural hematoma or a subarachnoid hemorrhage. And when you think about that, I think this is easier to draw than just to describe. Think about the skull as kind of being this line right here. Here's the brain right here. And your brain itself has, it's lobulated, right? It's got two cerebral hemispheres. They're divided into lobes. And from the side view, you have like your cerebrum, and then you end up having a cerebellum back here. So they're lobulated. And one way that we can kind of keep the brain in position inside of that hard bony skull is to add some protective membranes like meninges and form like hard physical partitions. So this way, if the head kind of shakes this side, the brain doesn't necessarily bounce back and forth and hit up against the walls of the skull. The meninges can hold the brain physically in place in between the two cerebral hemispheres. And we also have a portion of meninges that will hold the brain in place between the cerebrum and the cerebellum. So that keeps you from shifting forward or back too much or shifting side to side too much. So that's one of the basic functions of meninges. But there's not a lot of space in here. So we have fluid that's found in here as well. And if you go and you swell up any of these layers, or you go and add anything to this space that wasn't supposed to be there to begin with, you run into problems. So let's just pretend now that we're just looking at layers of the meninges. So meninges has three layers. You have dura mater, you have arachnoid mater, and then you have pia mater, right? And so a lot of times people just remember that DAP, arachnoid mater, pia mater. And then between that, you have spaces. So if it's between the dura mater and the arachnoid mater, you have the subdural space. So we'll just put SD. Between the arachnoid mater and the pia mater, you have the subarachnoid space. So imagine this is a very tiny space to begin with. But now I end up having like a big blood clot in here. You know, what's that going to end up doing? It's going to compress that space, and that might compress the brain. Or if I have an active bleed right here, that's going to take up space. That can cause compression of the brain. So either way, when you're adding something to that space that's not supposed to be there, it's no bueno. So sometimes you'll hear about subdural, what's called hematomas, or maybe a subarachnoid hemorrhage. And again, this isn't an end all be all definition of how these things form, but a common way that maybe a subdural hematoma could occur is if you do some type of coup or injury where the skull is moving and hits a stationary object, or maybe you have a moving object that hits a stationary skull and it causes the brain to kind of shift around inside the skull. That could cause the dura mater to tear a little bit. And if the dura mater tears, it'll bleed into that space, leading to a subdural hematoma. So most of the time, if somebody has a subdural hematoma, it was caused directly by head trauma. It was caused by trauma. So clearly they're going to have a headache, but they're also going to have a lot of neurological symptoms. And I can go over those with you a little bit later. But one of the ones I used to tell you about all the time, because I had that lady that came in and threw up on my shoes, is you might get what's called projectile vomiting, where they just kind of throw up and they're like, oh, I'm sorry about that. And if you remember that lady that used to come into my office, she's like, oh, that happens all the time. And I told her, I said, frequency does not make it better. But she had something going on in that space, and it was causing compression. So something was compressing the vomit centers of her brain, and that's what was causing her to get the frequent nausea and the frequent vomiting. So if somebody just kind of throws up without a warning sign, we sometimes call that projectile vomiting. It could indicate that they're not necessarily sick from like a bacteria or virus. I mean, they could be, but it's probably coming from some type of nervous system inflammation around the brain. But dizziness, drop attacks, diplopia, double vision, but you would have a headache and some other type of neurological symptoms to go along with that. So subdural hematoma can present with that. Usually that would follow trauma. Or you could get what's called a subarachnoid hemorrhage, where they'll usually say, oh, it's the worst headache I've ever had. It's getting worse. And that's usually coming from an active bleed. So if I have a blood vessel that ruptures and it starts to bleed out and fill in that space, that's usually what a subarachnoid hemorrhage is. So maybe you already had a pre-existing aneurysm and the aneurysm ruptured. So if that's a new concept or something that's kind of hard to conceptualize, an aneurysm is where you have a blood vessel that's ballooned out. And we'll talk about this more when we get into cardio, but again, repetition is the key. And when you have a blood vessel that's ballooned out, it's just weaker. So this has become distended out. It's been stretched, and sometimes a rapid change in pressure, like if your systolic pressure, it ends up being way higher than your diastolic pressure. It can put undue pressure on these already pre-existing aneurysms, and these things can dissect. And a dissected aneurysm just basically means it ruptured. So you would end up having an active bleed going on out here, right? And that blood's going to take up space. So sometimes that's how a subarachnoid hemorrhage takes place. But in this case, they don't necessarily have a pre-existing trauma. It's just something, all of a sudden they get a headache and it's getting progressively worse and worse and worse. And the reason it's getting worse is because you're getting more and more blood stuck in that area, right? So it's the worst headache of my life. They end up having other neurological symptoms, but they know it continuously gets worse. And then the other idea, which I don't think we need to use the picture anymore, is, well, what if the meninges themselves become swollen from infection or inflammation? That's where the term meningitis comes from. So itis means inflammation of the meninges. So maybe you have some type of bacterial infection that's caused it. Maybe it's a viral infection, maybe some type of fungal infection. But regardless, the meninges themselves will swell, and you've got a very small space in there. So any swelling causes compression, and when we feel pressure on the brain, that causes neurological symptoms. So in this case, the basic signs and symptoms, if you'll remember when we talked about meninges, was headache and nuchal rigidity. Let me see if I can find that on here. If not, I'll type it in for you. But one of the basic signs and symptoms of meningitis, so meningitis is nuchal, what's called rigidity. And that just means a really, really, really stiff neck. And it's so stiff you can't move it. And if you do try to move it, you'll probably flail a little bit. So that might have been something that I'd shown you a long time ago. So there's certain, you can either call them neurological tests or orthopedic tests, where we'll force flex the chin, and rather than looking for a limit sign, which we had just talked about with MS, where you have the sheep pain going down the neck, to see if by tractioning the neck, does that cause any type of response? And so sometimes what will end up happening is if we flex the chin to the chest, you'll see the legs will twitch or the legs will flail. They'll come up into a frog leg position. And then another thing we can do is we can extend the foot out and dorsiflex the foot, which will cause, it's almost stretching strings in a way, and it'll cause the leg to jump or the other leg to jump. And that tells me that I have tension in the nervous system. So with meningitis, again, you're going to get a headache. You're going to have definite nuchal rigidity. You might have a positive Kernig's or Brzezinski's test, where we're seeing that there's some type of cord or nervous system tension that's going on. And depending on where it comes from, would normally determine what the proper course of treatment would be. But because testing takes time, normally if you come in with signs and symptoms of meningitis, they're probably just going to go ahead and give you antibiotics regardless. So clearly, if it's coming from a viral infection, that's not going to help that much. You know, if you're not receiving some type of antifungal medication, it's probably not going to help that much. But what they're trying to do is make sure that we can treat it as if it's a bacterial infection. Because if it is, the antibiotics will go ahead and start working. And if it's not, it's almost like a no harm, no foul type thing, because viral infections usually get better on their own. So a lot of times you never know what it was unless they do a culture. But certain things they can do to test meningitis, I've probably mentioned this to you before. They can test the CSF and see if there's increased protein, which might indicate that there's an action going on. And usually increased protein occurs with infection because you're producing more antibodies in the blood, and that ends up creating more globular protein in the blood. But they might also notice that there's a decrease in sugar in the CSF if you have bacterial meningitis. So increase in CSF protein, decrease in CSF sugar might indicate that it's bacterial meningitis. And then, of course, they do a culture to see which type. Cerebrum, main parts of the brain. So it gets subdivided into two cerebral hemispheres. The hemispheres themselves, right and left, to get subdivided into lobes. So here we can see the different color lobes. So temporal lobe, occipital lobe, parietal lobe, frontal lobe. All this is going to be written out for us, but let's just kind of talk about it while we have the picture up. When you think about frontal lobe, Um, here, the prefrontal cortex ends up doing a lot of our planning, logic, um, higher cognition, planning difficult tasks. Short-term memory is up here. We have Broca's area in the frontal lobe, which is going to be the motor area for speech. That usually sits right about here. You have the precentral gyrus right here. The gyrus is your primary motor cortex. And then you also have an associate motor cortex up here. So plan movement, actually initiate a movement right here. And I'll talk more about precentral and postcentral gyrus in just a minute when we have a picture. But this is the part of the brain that, if it's in charge of most of your voluntary motor activity, imagine it's damaged, that would lead to certain types of paralysis, perhaps. So we can kind of look at this based off what does it normally do, or we could look at this based off of if it's affected, what would happen. So if part of the brain gets damaged, you're going to have a hard time thinking and planning and coming up with logic and understanding social norms and values. So this is an area of higher thinking. If this area of the brain got damaged, you would have difficulty with speech. You would have some type of aphasia. So Broca's aphasia is where you can't make words anymore. If the virus got hit, where we have voluntary motor function, then you're going to lose some voluntary motion depending on how much of it was affected, and so on and so forth. So when you think about parietal lobe is your main somatosensory portion of the brain. So it's picking up information from your skin and joints mainly. Occipital lobe is your primary visual cortex. It's where we're processing visual information. When you think about temporal lobe, smell, auditory sense, that kind of stuff. So you should be now for here. I've outlined them for you. Why is the precentral gyrus called the precentral gyrus? Why is the postcentral gyrus called the postcentral gyrus? Because the kind of the space that sits between my frontal lobe and my parietal lobe, there's a deep depression. So sometimes that deep depression is called a central sulcus. If I happen to be the lump in front of the central sulcus, I'm called precentral gyrus because the gyrus is a lump. Right? So central sulcus, that depression right there between the frontal lobe and the parietal lobe. If I'm the lump gyrus in front of the central sulcus, precentral gyrus. If I'm the lump behind it, postcentral gyrus. So this isn't the best picture to show that. This should be more of a continuous line. This one better. So here, precentral gyrus, postcentral gyrus, your central sulcus. So primary motor cortex. Here's the prefrontal portion of the frontal lobe. So prefrontal cortex, thinking part, voluntary motor, motor for speech. Parietal lobe back here. This is the main part that is picking up, again, information from your skin and joints. So we sometimes call this the primary somatosensory area of the brain. But precentral gyrus and postcentral gyrus is another name for it. I think sometimes people get a little bit of confusion with those. Everything's written out for you. Again, with parts, it's a little bit long, but it's pretty straightforward. Most of the time with the brain, thinking this part does this part does this, and this part does this. But sometimes you'll have certain areas of the brain that are a little bit more complicated than others. Like frontal lobe is going to be a little bit more complicated than parietal lobe. But everything's written out for you. Let's come back to cognitive orders in just a little bit. But the reason this kind of sits here is because, again, we're talking about the prefrontal cortex. One of the parts of it is thinking, higher thinking. So we'll come back to that in a little bit. Parietal lobe, again, somatosensory in nature. Again, somatosensory, I know I'm using that word freely. And again, we're trying to remember all this stuff from a year ago. Somatosensory are the things that we can consciously feel. So when you look at the pre or post central gyrus, you could subdivide them into different regions. So for example, if I look right here, you can see mouth, eyes, face, hand. So when you're looking at the sensory homunculus or the motor homunculus, what that's basically saying is this part right here, there's a certain part of it that's just in charge of the face. There's a certain part of this that's just in charge of the hands. So if I do touch my hand, for example, maybe that gets registered right here on the post-central gyrus. If I want to grasp a pencil with my hand, maybe that takes place here in this portion of the pre-central gyrus. So again, different parts of the brain are going to be responsible for different functions. But when you get into the pre-central gyrus and the post-central gyrus, these are further subdivided where each region is going to represent a certain portion of the body that it represents. So again, if this represents hand, that's going to help you control the hand. If this represents hand, that's going to allow you to feel the hand, like feel touch and itch and burn, pressure and vibration sense and all that kind of stuff. But again, somatosensory means you're consciously feeling things. Let's see. Another application of that, what if I got a stroke right here on, say, the left side of my brain? Then perhaps I'm going to have loss of the ability to maybe close the eyelid. But if the stroke is localized, maybe that wouldn't affect, say, the hand. So maybe depending on how large the stroke is. So let's go back to big picture and then we'll come back to small picture again. Let's say we have a stroke that affects this entire portion of the brain right here, then I'm probably going to end up losing the ability to control muscles on the complete opposite side of the body. Right. So maybe not necessarily in the face, but anything below that, I would lose contralaterally. So if the area gets affected by a stroke or brain damage, I might have what's called hemiparesis. I lose the ability to control half of my body. But if it's just a smaller area, like maybe a lacunar infarct, like a small stroke, where it's still a stroke, it's not a TIA. I'll try to go over the difference with that in just a second. But only end up affecting a portion of the body. Maybe I'll just droopy eyelid, or maybe I'll just have droopy mouth, or maybe I'll just lose the ability to move my hand. It really just depends on to what degree was that area affected, right? So let me go over a couple of words I just mentioned to you. These aren't necessarily testable for me if they're not in the notes, but I think you should be familiar with them just in case. Stroke usually implies that we've had some type of cerebral vascular accident in which damage is permanent or it's lasting longer than, say, a day. Most of the time, this isn't something that's going to resolve if you've had a stroke. It probably does imply a decent amount of damage has been done if they just use the word stroke. But you could also have what's called a infarct. And that usually just like a heat stroke, where a small or a smaller area of the brain has been affected. So there might be minimal damage, or maybe only one part of the body gets affected in a very small region, but it's still a permanent thing. So a stroke is an infarct of brain tissue, but it usually implies that maybe something large has taken place. A lacunar infarct might be something like a mini stroke where just a smaller area has been affected. You can also have a TIA, which is transient ischemic attack. These typically have stroke-like symptoms, but they tend to resolve within like 12 to 24 hours. So that's where the term transient ischemic attack comes from. So why are they using the word ischemia to begin with? Remember, ischemia we talked about in heart discussions. Ischemia is where you have too little blood flow or too little blood supply to metabolic demand. So let's say that's temporary and the blood eventually returns in time, then it's a transient ischemic attack. You had ischemia, but eventually the blood returned. If it's a stroke, it's a full-on infarct, right? A stroke is an infarct of brain tissue. So the difference between the word ischemia versus infarct is an infarct is prolonged ischemia. and tissue damage occurred, like permanent tissue damage occurred. And of course, a TIA could be very small, could be very large, right? But sometimes you'll end up hearing like a stroke versus a mini stroke, and sometimes it just depends on which vessel got affected. Was it a big vessel? Was it a small vessel? Was it a big rupture? Was it a small rupture? And then you'll sometimes hear differences between the word stroke versus transient ischemic attack. This will get better. This won't. Right? You might get improvement, but it's never going to go back to the way it was originally. And really, what's implied with TIA is it goes back to normal. So maybe if it doesn't go back completely to normal, it wasn't necessarily a TIA. Let's see. And I know this seems disjointed, but I'm trying to go over big concepts with you too, because ultimately we don't want to just understand a bunch of words. I don't want to understand what it all means and how we use it one day. Just read through this, just know what different parts are responsible for. Wernicke's area, sometimes they'll put it in a singular place. I think I like this picture a little bit better. Wernicke's area is not in any one lobe, but actually is a communication point amongst multiple lobes, because we sometimes think about Wernicke's area as our sensory area for speech and communication. So whether I'm reading written language or I'm understanding spoken language, there's a lot of inputs there. Like I'm listening to something, or I'm seeing something, or maybe I'm even, watching somebody do sign language. That would also have to do with language and communication. So Wernicke's area is receiving that information. It's understanding that I'm hearing spoken language or I'm reading written language. And it helps you understand the syntax, the grammar. Broca's area, again, was back in the frontal lobe, and that's motor for speech. But if I have a problem with either area, that can lead to different types of aphasia. So you will probably experience this sometime in your life with your patients. And sometimes you'll hear somebody say, this patient has become aphasic. So that means they're having difficulty with communication. And again, it might just be temporary. Maybe they had head injury and they're going in and out of aphasia, or they're becoming aphasic. But it certainly should point to maybe there's something going on in the skull here. Maybe we should take an image, right? So if a patient is going in and out of aphasia, certainly there is something going on in that brain that we might want to look at. So there's something causing pressure on Wernicke's area. If they're just saying words and they're not making any sense at all, or maybe something's going on in Broca's area if they just can't talk to you anymore. Right? And again, there's other things that could lead to that, but that's an idea. Let's see. What else do we want to talk about? Occipital lobe, visual interpretation. I'll talk about the optic chiasma when we get to neurosensory, I think. But that is a good chart right there for, if you remember when we talked about something called hemianopsia. Hemianopsia is where you have partial vision loss. Here you have vision loss, and partial vision loss. These are partial vision loss. Like I've lost the lateral eye field. So these are sometimes good indicators of where a problem might be located. So if it ends up being in the optic chiasma, a lot of times we end up getting hemianopsias. If it's just centered in, say, the optic nerve, then I might have a complete anopsia, right? And then sometimes you can even have something in the occipital lobe that would lead to a different type of hemianopsia. Now, let's not skip over this for right now. This way it'll save us time when we get to neurosensory. Let's go ahead and talk about it now, because this is a good image, and I don't know if I've carried this over to the neurosensory portion. But sometimes when we learn neuro and we get very decent at it, we can get what's called localizing lesions, right? Because if you understand how everything's connected, it makes sense that I can see a normal vision or have normal visual acuity. If you were to show something in front of me and I were to read it back to you, there's a lot of things that are working there. My retina of my eye is working. My optic nerve must be working. The connections between those nerves and the occipital lobe must be working. The connection between my occipital lobe, my Wernicke's area, and my Broca's area must all be connected. So if I'm reading a written word to you, that means all the connections are complete. But what if I were to ask you to do an eye chart and there was a part that you couldn't see, so I end up doing like maybe some type of cardinal fields of gaze, or maybe I end up doing some type of test and I notice that you say your peripheral vision or your central vision. That can lead us to different types of diagnoses. So for example, there might be a picture that you remember of me drawing that looks something like this, where we had, like, say, an eyeball, and let's pretend we're looking down on the top of a head, and the eyes are looking forward. So let's say this is my left eye. Here's where my nose would be. Here's my right eye. Here's my visual field out here, like what I'm seeing out in front of me. And then on the back of that, of each eye, you have a retina. That's the light sensitive portion inside the back of the eye that will consolidate its information down to an optic nerve. The optic nerve is going to come back, and right around where the pituitary gland is, you're going to end up forming this thing called an optic chiasma, because it looks like a little X formation. And from there, it's going to continue on to the into the process. But ultimately, everything has to be connected to work. So if I, for example, have to, or if I'm trying to determine where a lesion might be located, imagine I were to cut this right here. Well, this is my entire right eye, entire left eye. Everything that I was trying to process from my right eye will not make it. Therefore, I will have right-sided anopsia. I will not be able to see from the right eye. So that might on a chart look something like this. And let's not flip the chart around to where it's facing you. Let's just say that this would be blind. So that's anopsia, like I cannot see, right? So anopsia, opsia, put a in front of it, anopsia, cannot see. But one of the things we had talked about a long time ago is, well, what if this part gets affected? You know, that wouldn't necessarily create anopsia. I wouldn't have an entire eye field that's affected, because when you get back to the optic chiasma, if you remember, that's where exchange takes place between the medial eye field and the lateral eye field. And we get some crossing over that occurs here. So let's kind of focus on that part a little bit. Now you have an optic nerve coming back. I'm going to draw this kind of wide now. And here's the other optic nerve coming back. And I'm just going to kind of draw that a little bit more straight. If your information coming from, say, the lateral eye field versus the medial eye field, that will determine which occipital lobe you go to. So here's a lateral eye field, that's a medial eye field. Lateral eye fields always stay ipsilateral when they go to their occipital lobe. They stay on the same side. But the medial eye field always crosses over. So this will go that direction, okay? Again, lateral eye field is going to stay ipsilateral, but medial eye field is going to cross over. So you can literally follow these things if you wanted to. And if I ended up having a problem with my optic chiasma, then that is a problem that looks more like, say, this or this. And those things are called hemianopsias. So let me show you why. If information was going from, say, let's just do one example. Let's say we have an optic nerve. Here's another optic nerve. Here's the optic chiasma. Let's say we have a pituitary tumor right here. It's affecting this part of the optic chiasma. That might end up affecting information coming from this field right here, but it might not necessarily affect this, right? As this comes down through here, this might get affected, but it might not necessarily affect this. All right. So what would that end up looking like? Well, this part right here, it made it on this side, so that part's still clear, but this part is now missing. And similarly, on the other side right here, this part didn't make it on the middle side, but that part did. So that's a type of hemianopsia. And only at the optic chiasma could that form of hemianopsia have taken place. Right. I don't want to get too deep into this because the type of hemianopsia change depending if I'm in the occipital lobe and I have a lesion down there. But you can see why we only have partial vision loss if it's the optic chiasma that's affected versus if it was the optic nerve. If it was the optic nerve that was affected, this entire eye would have been lost. But if the optic chiasma has been affected, a portion of this was lost and a portion of this was lost, but not either one completely. Okay. And so what I'm not trying to do is teach you how to learn to read a hemianopsia chart. What I am trying to teach you is understanding why lesions are important in the nervous system because they interrupt flow. And if you understand where the flow is supposed to go, you could probably localize the lesion if you got really good with that. So that's kind of what we're doing. Like when we're learning different parts of the brain and we learn what it does, let's say we have a deficit in speech. So if I have a deficit in which I can no longer talk, but everything else seems fine, then I'm probably going to guess that my problem is somewhere right in here. Right? So let's say everything else is fine, but the person can't talk anymore, then I'm assuming Broca's area is malfunctioning, right? There's a lesion, maybe it's permanent, maybe it's temporary, but for whatever reason, this person, he was able to talk yesterday, he was able to talk an hour ago. Right now, he cannot talk to me, he cannot make words, seems to be able to listen just fine. He's, he can understand what you're saying, but he can't talk. So where would the lesion be located? So that's a lot of times what we're doing is we're trying to get good at neuro so we can localize lesions or at least know where to look in general. That's one of the gists of neuro. Let's see. Most common reason for hemianopsia that affects the optic chiasma is the pituitary tumor, and it's just because they're anatomically located close to each other. Nuclear basal ganglia. When these come up, the reason they're probably going to come up is because you have somebody with some type of movement disorder, such as Parkinson's disease and Huntington's disease, or you could call it Huntington's chorea. Chorea, it's not just Huntington. You also have what's called Sydenham's chorea, and there's various other different types. Parkinson's also has other diseases that look similar to that might be more drug induced. So they end up having Parkinsonian-like movements. But let me give you a general rundown about basal ganglia one more time. Let me show you some of the key features of Parkinson's disease or Huntington's disease one more time. And I think that should suffice again. But basal ganglia, one of the functions that they do, they help initiate. They're like, let's get this going. Let's get this moving. They initiate movements and they help to have the right amount, if you want to say it that way. So to make it not too much or not too little, we sometimes call that modulation. Like we're modulating, like you're tuning something up or you're tuning something down. So when it comes to skeletal muscle movement, there's two main neurotransmitters that kind of tune it up. That one's called acetylcholine, and one that tunes it down, which is called dopamine. So again, there's other uses for acetylcholine other than skeletal muscle movement. There's other uses of dopamine other than skeletal muscle dampening. But in the case of using skeletal muscles, acetylcholine amplifies and dopamine de-amplifies it. It tones it down. So in order for our muscles to work appropriately, the basal ganglia need to initiate them, properly modulate them, make sure there's the right amount of amplitude or toning down. If, however, we have a problem with basal ganglia, then you have difficulty with getting muscles started. So a person has difficulty getting moving. Like they might have a gait, or when they walk, they might have to lean forward a little bit just to get going. And when they're at rest, they tend to move too much. When they're trying to move, they tend to be too rigid, or the movement is too little. So again, that's a improper initiation, and then that's improper modulation. So one of the most noticeable things about Parkinson's disease and Huntington's disease are things called resting tremors. They're called resting tremors. And they look a little bit different in Parkinson's versus Huntington's. So it's hard for me to show you just on like a doc camera, but Parkinson's usually looks a little something like that. It's just a repetitive motion. A lot of times the hand, the thumb might be folded in like this. Sometimes it's facing this way, sometimes it's facing that way. But the hand, when they're resting, shakes. And it's a repetitive type of motion. With chorea, it rides. And you might notice this more in their face than their hands, but it'll affect the hands too. So with chorea, when they're at rest, you just notice they're all squirmy. We call that a writhing type of movement. With Parkinson's, it shakes when they're at rest. So Parkinson's looks more like that. Okay. And of course, it doesn't just affect the hand. It might start in the hand, but it tends to go on to affect the face and other parts of the body as well. But again, that's when they're at rest, they tend to have what's called resting tremors, right? They shake, or their body will start writhing. So I might have some videos. Let me see if I got any embedded videos of this. Here we go. Here we go. So here he's having difficulty getting started. Hands are shaking a little bit. So the hands, when you're in Parkinson's, the arms don't tend to swing back like normally would. So his hands are at rest because the arms are stiff. So that you can see the hand shaking at the end, and at the beginning he's having difficulty getting going. So they sometimes call that a Parkinsonian gait, or they sometimes call that a festinating gait. And then here, this is the classic, what's called pill rolling tremor over here on the right. And the reason they call it a pill rolling tremor is because it almost looks like you got something in your hand that you're kind of shaking around. Like if I had like a little marble or a pill in my hand, it looks like I'm shaking it around, like keeping it moving around in that little area right there. So that's why they call it that. So that tends to be very early. And like I've shown you before with some of my patients, they just had something like this. It was hardly noticeable at all, but that's how it starts right there. You'll just start to see that hand while they're just sitting there doing nothing. They're not really paying attention to it. You'll just start to see them do like that. And that's real early Parkinson's. Parkinson's is a lot more prevalent now. One of the theories behind that is some of the fertilizers that we're using in the pesticides. So much more prevalent where I used to live up north, up in Indiana, because I mean, you're surrounded by that stuff all the time. There's huge fields of corn and soybeans, and you're constantly spraying fertilizers and pesticides everywhere. And even it's not the pesticides, but it can even be the glyphosates, like the weeds, the herbicides, I guess you should say. So those things are starting to get more and more tied into some of these movement type disorders and these CNS disorders. So I had, I think, 1, 2, 3, 4. I had 5 neighbors within visual range of my house. 4 of my neighbors had Parkinsonian type conditions. Let's see. some of the terminology associated with Parkinson's. The problem, again, is it's a basal ganglia disorder. We're not producing enough dopamine, so that's going to end up leading to the logic of the treatment that we use for it. But the patient's not able to properly modulate function. Signs and symptoms: resting tremors, repeated movements approximately 4 to 6 hertz. That's just how often the movement, like how often is it shaking, like per second. So when I'm shaking, you know, how often per second is it going back, like 4 to 6 times per second. Starts off with a pill rolling tremor. Other signs and symptoms include bradykinesia. Bradykinesia is like slow, stiff movement. You see where he's like hunched over there, very stiff. And again, when they walk early on, you might notice that they stare. It's a really blank stare, and the arms don't move at all. Like they just seem stiff to the side, but they don't have the natural swing to them. The one he's showing here is pretty advanced, and it would probably be just before they're wheelchair bound, because eventually they will become wheelchair bound and almost completely stiff. But he's showing what's called a festinating Parkinsonian gait. You become increasingly rigid, stoop posture. He was showing that. Difficulty swallowing. Obviously, more muscles get involved. So if I can't properly swallow, a lot of choking incidents, falls, soft voice, whisper. So I had a lady. She was quite advanced, and you really would have to lean down to listen to her talk. If you wanted to get her up on an examining table, you physically had to pull her out of the wheelchair. You physically had to make her more erect. Like she was completely hunched over, and it didn't hurt her or anything like that. You felt like you were hurting her, but you physically had to stretch her out to get her to sit up. But you had to lean over all the way to her mouth to hear her whisper. Treatment, like I was mentioning before, levodopa, you want to bring back the dopamine. Because when you think about things like the bradykinesia and you look at him, the muscles are too tight, too active, too hyperstimulated. They're rigid, right? The muscle's too tight. Here, it should be at rest, but it's still active. So how can we dampen some of those effects? You need that dopamine to dampen the muscles. So if I don't have enough dopamine, the muscle is getting too intense a signal. They're modulated too high. So you become very stiff and rigid, which slows your movements down when you are trying to be active, and you get that shake when you're not trying to be active, right? So the resting tremors. So that helps explain why dopamine is the treatment. Let's see. What else? The Huntington's chorea. This one has a different appearance. So with Huntington's disease, again, we end up getting more what's called writhing type movements or athetosis. Some people call it snake-like movements. It looks like a snake or a worm is squirming. Oops, sorry about that. Slow eye movement, dysphagia, cognitive disorders, and depression. That does later. Issues, difficulty swallowing again. Here we can see the walk. It might not be that pronounced. And one thing I want to kind of point out to you, if you have ever run across Parkinson's patients, they might also have the same walk when they're being medicated, right? So this is a non-medicated Parkinson's patient. This is what a non-medicated Huntington's chorea patient might look like. But this is also what a medicated Parkinson's patient might look like, because sometimes some of the effects of the levodopa causes hemiballismus, where you start to flail like one side of the body versus the other. So they might have like a blank stare, but you might notice that the body starts flailing in some of these arms, right? So sometimes the look more rigid. When you treat Parkinson's with levodopa, they tend to start having more of a flailing type of movements, so they look as though they have chorea. But here you can kind of see that dancing type motion he's got with chorea. And I'm not sure if this is Huntington's or Sydenham's that either one of these have. But again, that word chorea comes from the word dance, like choreography. So it looks like they're dancing. but both are basal ganglia disorders. Thalamus, epithalamus, hypothalamus. Thalamus, we always remember as the sensory relay center. Epithalamus is kind of like more of a region. So you got the pineal gland there, which helps with day-night cycle monitoring. It helps set your biological clock. Problems with gland could lead to problems with sleep, eventually mood, because we need melatonin to sleep. And during the day, melatonin gets converted to serotonin, which helps you feel good. So sometimes if people have pineal gland problems or they work like, say, third shift, they get seasonal affective disorder. Or if you don't get enough sun exposure, you can get seasonal affective disorder. But thalamus sensory relay center, a lot of information comes into the thalamus. The thalamus redirects it where to go. Bigger thing to get inside those hypothalamus, of course, because the hypothalamus is in charge of homeostasis. So this is the part of the brain that I consider the most important part of the brain. Like, it sets most of our homeostatic set points. It's in charge of the autonomic nervous system. It's largely in charge of the endocrine system. This is kind of the thing going. So if a patient ends up having pressure on their hypothalamus, they're going to be all messed up. So imagine somebody with a head injury and they come in and their body temperature is fluctuating like crazy. Their blood pressure is fluctuating like crazy. Their pH is all messed up. Their breathing's all messed up. And that could indicate a brainstem issue as well. But everything that's supposed to be homeostatic regulated is fluctuating like crazy. That could tell you that there's pressure on their hypothalamus. So, but again, basics, hypothalamus is largely in control of homeostatic mechanisms. So, for example, if I want to maintain proper body core temperature, we have to have something sensing it, of course, which is the thermoreceptors. But I have to have something that's saying, well, this is where we need to be ideally to begin with, right? So hypothalamus is the one that's the control center. It's the one that says, I like 98.6 best. If you're at 98.1 or 99.1, I'm not upset with you. But if you go below that or above that anyway, it's not going to work. So again, that's what the control center for homeostasis does. It's the one that's setting your ranges. And then it can also end up communicating with somebody that can do something about it as well. So the hypothalamus might say, all right, well, we need to fix this. So you need to shiver more, sweat more, breathe fast, breathe slower, increase, vessel tension, all that kind of stuff. So it can communicate with other parts of the brain that can make effects. But again, the control center for homeostasis is the one that's kind of controlling all your set points. So you can imagine that if something's wrong with the hypothalamus, all your set points are going to be all messed up, right? So people are going to wildly fluctuate in temperature, pH, osmolarity, everything gets messed up. Let's see. Corporal quadrigemina we've talked about before. It's 4 little lumps. So again, I think it's just easier to draw it. Corpse means body, quad 4, Gemini, like twin. So if it's a corporal quadrigemina, it means 4 twin bodies. So you end up having this little hill, superior colliculus, followed by another, an inferior colliculus, and then one's going to be on the left side and the other one's going to be on the right side. So what do I have now? 4 twin bodies. So all together, that's called a corpora quadrigemina. But again, it's made up of a superior colliculus up top and inferior colliculus down at the bottom. And superior colliculus is what kind of ties in eye movement with like the neck and the body. So like if I'm following a moving object with my eyes can follow to a certain point, but eventually I need to coordinate neck movement and body movement to keep following it. Like once you get out of the field of vision, I'm going to have to turn my body or turn my head to keep following that moving object. So superior colliculus plays a big role in that. Inferior colliculus is what allows you to turn toward a sound. So there was an interesting study when they're working on these like quantum computers and stuff, you have to have a very vibration free like facility to do that. So they make these super quiet rooms, and they were noticing that it actually affected people's balance, because one of the ways we actually balance our body is by being able to hear like where we are in space. And you don't think about this from a normal sense, because there's a lot of other senses that are kind of superseding, like the how much noise is bouncing off a wall or a floor to me. But they started noticing that when they put these people in these super quiet environments where there's hardly any sound waves at all, because they all get absorbed, they're actually losing their balance, and the inferior colliculus is getting affected. But big application. The better my superior colliculus is, the better I would have like body-eye coordination. So sometimes birds of prey end up having really large superior colliculi. And then things that tend to use sound as a way to guide themselves, they tend to have really large inferior colliculi. So bats and owls have really large inferior colliculi. So in a bat, it might look more like this. They're not really proportionate. In an eagle, it might look more like that, right? It just depends on what you use more. Ours, the superior colliculus is a little bit bigger than the inferior colliculus. But together, you got these 4 little twin lumps, so they call it corpora quadrigemina. When they get into cranial nerves are ordered from what's called rostral to caudal. So kind of the further you go back in the brain, the number goes up. So by the time I get down to the bottom, I'm kind of like at 11 and 12. So here in the midbrain, you had exiting cranial nerves 3 and 4, which is oculomotor and trochlear. Pons and medulla make up the brainstem. When you get into pons and medulla, here's a decent picture of that. The pons is the portion that kind of sticks out. It's got the little lump. The medulla is another portion where you do kind of see like a little lump on it, and then it starts to taper down and narrow. Once it kind of forms like a smaller portion down here, and now you're at spinal cord. So this is like the last little portion between your brain and your cord. So that's why it's called a medulla. It's in the middle. So if cord's right here, and let's say the brain is all up here, the medulla is right in the middle. So that's where the term medulla comes from. But together, pons and medulla make up the brain stem. And brainstem is in charge of basic vital function. So breathing takes place in both pons and medulla is kind of solely in charge of cardiac function, right? So damage to pons and medulla is no bueno, right? You're going to have difficulty breathing, maintaining proper breathing rate, blood oxygen and blood carbon dioxide saturation rates are going to be all off. If you have a problem with the medulla, heart and blood pressure will be messed up. Okay, so brain stem, very important, just a lot, right along with the hypothalamus. These are some of the 2 other very important parts of the brain just to keep you alive. So you got the hypothalamus that kind of sits right here, and that is maintaining proper homeostasis. And then you have the brain stem, pons and medulla, that's for respiratory or pulmonary control and cardiac control. Let's see. So there's pons, medulla. Cerebellum is for muscle coordination. So lack of coordination, if you remember, we call ataxia. So if you see a person that looks like they're drunk, or they're moving around like they're drunk, they could be drunk, or they could have some type of cerebellar issue. So sometimes people with MS have that drunken ataxic gait. I think this guy is compensating for that. Hers non-compensating. What that means is this is how she normally walks. So she has some type of cerebellar ataxia going on, probably MS that's caused this, or cerebellar tumor, perhaps. He also has a drunken ataxic gait, but what he has done, if you notice, his gait has gotten wider and his toes are pointing out, so he's compensating for that. So that way he doesn't get as much crossover as she gets. You're not really seeing the crossover much, but if I put a little piece of tape on the ground and I tried to watch her walk along that piece of tape, her feet would cross over. Right. But again, she's slightly compensating. He is largely compensating by widening that base and pointing those toes out. But again, he looks drunk, right? So again, most likely this is probably MS. It's doing that. It doesn't just affect walking. If you have cerebellar problems, you're also going to have difficulty hand-eye coordination. A lot of times you end up getting what's called intention tremors, which kind of go with dysmetria. So you've probably seen me do this before, but if I'm going to reach something, you might notice I shake when I'm going for it, or I get real clumsy when I try to do it. That's different than Parkinson's. Parkinson's, when you're sitting at rest, you shake. With MS or cerebellar tumors, you shake when you try to do stuff, right? I really shouldn't have done that because now I have to go and get it. But it's called an intention tremor. Give me a second here. But also, let's see what we got here. Let me see if I went in any deeper. I can give you the words. If I didn't post them anywhere for you, I might write them down for you. Yeah, here's some of them. So dysdiadochokinesia, ataxia, nystagmus, intention, tremor, scanning speech. We mentioned that one earlier. One that I've mentioned to you before that I don't see on here. There it is. So dysmetria. I did write that down for you. I have mentioned that one a couple of times. The word dysmetria just means difficulty, like measuring something, and normally it's like distance and trying to do hand-eye coordination. So if you've ever seen a test where maybe the physician says, touch your nose, now touch my finger, you'll touch your nose and then you try to touch their finger, and then he'll say, touch your nose and now touch my finger, and he moves it. He'll usually move it closer or further away. And he's looking to see if that hand changes, because you might get it right the first time. But then when they start to move the distance or change the distance, he might notice that you start shaking, you have that intention tremor, or you miss it. And that shows signs of dysmetria. It's almost like you have poor hand-eye coordination when you have dysmetria. The dysdiadochokinesia, that's where you have difficulty with rapidly alternating movements. So you can YouTube this if you want to, but a lot of times the test for that is you just have them take their hand sitting on top of their other hand, and you have them flip it like this back and forth. And if they can't do that very well, then that's called dysdiadochokinesia. So sometimes, and I know this is really bright and flashy because of the camera, but they should be able to go pretty quickly. And if they can't do that, what ends up happening is the other hand ends up flipping on top. They just have difficulty doing that step. So the other hand ends up flipping on top. But you could probably YouTube dysdiadochokinesia, and it would show you a positive versus a negative. We already talked about Charcot's triad. Again, MS, we'd already mentioned before, typically affects the cerebellum first. So that's why a lot of this terminology seems familiar. Dysarthria is, it's another way of saying difficulty with speech. So back here, we talked about scanning speech or staccato speech. So here they're just being more generic. It's more generic. Dysarthria just means difficulty speaking, but it's not describing it in any way, shape, form, or fashion. It's just difficulty. Limbic system is the connection between, say, your hypothalamus and partially your sympathetic nervous system, if you will. That's one thing that we use it for. Imagine this. I smell a certain smell and it causes me to relax. Or I think of a memory as a child where I had fun and I was playing and it makes me feel good. And again, I relax. Versus if I think about the person in this world who I hate the most, all of a sudden I feel tense. My heart rate increases. You can feel your blood pressure going up. All that's occurring because of the limbic system, in which things like thoughts, memories, smells, fear, stress and anxiety can actually lead to somatic changes in the body. That's limbic system that does that. And the reason the limbic system has these connections is because the hypothalamus is part of the limbic system. And very close by to the hypothalamus, you have things like your amygdala, your hippocampus, your olfactory cortex, and things like nucleus accumbens. And sometimes when things are located close in proximity in the nervous system, stimulation of one could similarly lead to stimulation of another. So, and of course, they're all interconnected. So, for example, if I stimulate my olfactory cortex with a pleasant smell, that might remind me of a time where I was very happy, and that can cause me to relax, right? Maybe cause some vasodilation, maybe relax the vessels a little bit. Contrarily, another example I sometimes give you, you smell a very pungent, disgusting odor that might make you very tense, right? So again, there's a lot of interconnections, but if we just go individually, like what are the different parts of the limbic system and what do they do? Again, hypothalamus ties into homeostatic function. It's our control center for homeostasis. The amygdala is what we think about with emotions and drives, and it more strongly responds to fear and punishment, but it also has some ties into happiness. But usually with the amygdala, think about this as being, I don't want to do this because this is going to hurt, or I don't want to do this because this is unpleasant. That's different than nucleus accumbens, which says, I do want to do this because it's good, it's pleasant, it's nice, right? So you kind of have, again, like the angel and the devil on your shoulder, if you will. That's kind of like these two things, like, ooh, I want to do this, but maybe I don't, because if I do, I'll get punished, right? So there's kind of like a dichotomy there going on. Hippocampus is where we store long-term memories. And then the olfactory cortex is, again, where we process smell. And again, they're closely related to each other. They're closely organized right alongside with each other. So sometimes smell can remind you of something, and that can create a strong emotion, which will change the way your body reacts, right? But one of the reactions that you're going to have right before the test is fear. Like, if I don't do well on this test, then my life is over, right? So all of a sudden your heart starts speeding up, your respiratory rate starts speeding up, your blood pressure goes up. So yeah, limbic system. Reticular formation, it's you can kind of see here where there's different tracks, but they're interwoven fibers. Sometimes when things has what's called a reticulated pattern, it has this crisscrossing network. So what's happening with the reticular formation, bigger picture wise, is you have these incoming signals heading up to the brain. And let's say that the more they are, or the more important they are, it can cause the brain to be more alert or pay better attention to something. And of course, there's cognition involved with that as well. But because of the way the reticular formation is able to work, the more it's stimulated, the more alert we are. And it's able to pick out more important stimulus from less important stimulus. So again, cognition gets involved with that. Like, I would have to determine in my prefrontal cortex what I thought was more important. But once I do that, my reticular formation can say, okay, I'm going to let you through because we determine you're more important, and I'm going to inhibit these guys from getting through because we're not worried about them right now, right? So it can kind of help you focus a little bit better. So two things the reticular formation does. It stimulates the brain, or makes the brain more wakeful upon more stimulation, and it can selectively kind of pick and choose important information from lesser important information, like what's going to get in and what are we going to kind of not pay attention to. So certain things that can stimulate the reticular formation are things like light and sound. So if you want to be more awake, more alert, more light, more sound. If I want to be less awake, less alert, let's turn down the lights, let's turn down the sound. That's an application of that idea. Um, Before we get into seizures, let me go back up real quick. Let me go back up because we just mentioned cognition, and one thing I did skip over were some of the cognitive disorders. Um, Cognition, kind of think about this as higher thinking, reasoning, planning, logic. These are things that occur in our prefrontal cortex. There's actually a very specific part of your prefrontal cortex called the dorsolateral prefrontal cortex that is in charge of higher thinking and cognition. And over time, again, maybe if I have, like, say, type 3 diabetes, or if um, the brain is just getting poor circulation for whatever reason, we can run into certain types of cognitive disorders. So a lot of times these will occur with age. And dementia is a word that we associate with a pretty common type of cognitive disorder, in which maybe we're having difficulty memorizing things. Maybe it could be people's names. It could be, how did I get there? What's this place called? But it eventually can progress, and you might get lost in your own house. I used to have a neighbor up in Indiana, and he had some pretty advanced dementia, and he was the nicest guy in the world. So he'd walk over in his overalls and, say, what you working on? And I would talk to him about it. And then he'd tell me a story. And then he'd say, okay, now how do I get home? And I would just point him in the right direction. He'd start walking, he'd go home. And the next day he'd come out, and he'd come and talk to me, and he'd say, what are you doing? I'm like, I'm just working on this, cleaning out the garage or something like that. And then he would tell you a story, and he would think he's telling it to you for the first time, but it was the exact same story he told you yesterday. And he's like, okay, how do I get home? And then I'd point him in the right direction. And then sometimes by the time he made it to the front yard, he was lost again. And I could just see him start to wander off, going toward the other house. So I'd have to go and say, hey, you live right there. That's your front door. So dementia starts off kind of slow. It eventually progresses until they become fully dependent on people. It says in the notes, look for the 3 A's, like apraxia, agnosia, aphasia. So again, if I have difficulty with tasks, if I have difficulty remembering things, if I have difficulty even speaking and stuff like that, that could be signs of cognitive disorders. The probably most common one you're going to run into, and again, this is probably going to accelerate quite extensively in the near future, would be Alzheimer's, where you might want to add in attitude to that one as well. So they might have difficulty with tasks and remembering names and places and talking to you, but they also might get a little angry about it as well. But forgetfulness, of course, is a extreme sign of Alzheimer's. With Lewy body dementia, they'll also take all of this, and maybe you're going to add in some hallucinations with that as well. So with Lewy body dementia, hallucinations might be the key word that's going to separate one form of dementia from another. So Alzheimer's will probably be the most prevalent type, but you take the Alzheimer's signs and symptoms and you add in hallucinations, and now they want you to have a different diagnosis. So you add in Alzheimer's type symptoms with hallucinations, you have Lewy body dementia. Delirium, different from dementia. Delirium is usually something that it can come and go, but it's not one of those things that slowly progresses over time. It's usually within seconds, minutes, hours that something develops. So very rapid onset, could be anything from hours to days even, but just rapid onset of confusion. Maybe forget where they are while they're sitting in the room. If this occurs at a certain time of day and it only gets worse at night, we usually change the name of it. It's usually called sundowner syndrome at that point. And if you've ever met somebody with sundowner syndrome, it's the wildest thing you'll ever see. It might be almost to the second, to the minute, every single night at 5, all of a sudden the lights just turn out. They'll start standing up, and you'll be like, what you doing, Margaret? And she's like, I'm going to go and see my husband, and her husband's been dead for 25 years. And you're like, okay, well, have a seat right there. And she's like, okay. And then she'll start watching TV or, you know, knitting or doing her normal thing. And then 5 seconds later, she's standing back up again. And you're like, hey, Margaret, what you doing? Oh, I'm just going to go see my husband. And you're like, well, he's not here right now. Why don't you have a seat? And they're like, okay. And it's wild. But again, leading up to that, maybe during the daytime, they might seem mostly coherent, mostly normal, able to interact, able to talk, able to remember things. But as soon as a certain time at night hits, all of a sudden they have that rapid onset of delirium. That's more specifically called sundowner's syndrome. So sometimes sundowner syndrome might just indicate you have inflammation of the brain. But sometimes it is one of those progressive diseases that comes with like cancers, like brain cancers and such like that. Let me see if there's anything else I should go over with you. But yeah, consistently wants to get up and walk around, no real goal in mind, or the goal is just maybe nonsensical. They like to pace a lot. The problem with this, though, is sometimes these patients that want to get up and walk around aren't the most stable people in the world. So they end up becoming, again, fully dependent on nursing care or family members. So they become what we call full care patients. Like they need 24/7 monitoring. It gets pretty rough. Memory loss, confusion, paranoia. Nothing new to really talk about there. Let's go back real quick. If delirium is happening in a person that you would not expect it to occur in, probably most likely is they have some type of maybe dehydration electrolyte imbalance, or they just recently changed their meds. So I wouldn't be horribly surprised to see delirium in some of my older patients, especially if they were getting like some type of cancer treatment coming and going. But I would be surprised to see it in maybe like a 20-year-old, fairly healthy young person. So if that's the case, I would probably look for other signs and symptoms rather than a neurodegenerative disease. So could be that they didn't properly hydrate, and so they might have some type of rapid onset of electrolyte imbalance. Could be, I mean, maybe they even have some type of acute kidney disease that's causing that, or they might have recently changed their meds. That might be a question you'd want to ask them. All right, have you recently changed one of your prescriptions? Have you recently changed a dosage? It could be something like that. So again, sometimes these things are expected in the elder community, but what would make something like this show up in a younger person? Look toward those. What else do we have? Did we get to seizures? Okay. Let's come back to seizures. We'll do that last. I think one of the last big concepts I wanted to talk to you about was, let's do tracks real fast. I don't think anybody's going to have difficulty with spinal cord cross section again. Reflex arcs. Again, reflex arcs are just good at showing interconnections between CNS and PNS. And your cord is part of the CNS. There is some activities that the cord is responsible for. We usually call those things reflexes, like deep tendon reflexes or withdrawal reflexes. But most of the time, the cord ends up acting as a conduit. So sensory information comes to the cord. From the cord, it might go up to the brain. From the brain, it goes back down the cord and then out to the appropriate part of the body. And you know how we determine whether something's the appropriate part of the body is like, well, where do I want to control it? So for example, if I want to control maybe my leg, I'm probably going to exit the cord somewhere down here, right? If I want to control, say, a bicep or a tricep, I'm probably going to want to exit the cord somewhere up here, right? So obviously it would be a proximity thing. So if I'm trying to control upper extremities, I'm going to be exiting somewhere that's going to contribute to maybe a brachial plexus. If I want to control the lower extremities, I'm going to be contributing into the lumbar plexus or the lumbosacral plexus, right? So again, I didn't want to dwell on this too much, but I think it's too late for that. What the cord demonstrates to us is it is a part of the CNS. And what kind of determines whether something's part of CNS or PNS is physical location and, in a way, ability. So with CNS, kind of think about this is the place that's receiving information, processing information, and controlling things. When I think about PNS, it's more or less the connection between the CNS and the rest of the body. So it's bringing sensory in to be processed, and it's carrying motor out to effectors, right? So with the cord, you can kind of do both. If the cord's making a determination and sending an action out, it's usually doing that through some type of reflex arc. And that's, again, one of those things that we drew pretty consistently back in Anatomy 1, where you'd have some type of sensory afferent information coming into the dorsal horn. From the dorsal horn, it would immediately exit the ventral horn, go out to a muscle, and you were able to respond to a stimulus. So for example, if I touch something sharp here, right, I feel that it goes to my cord along a peripheral nerve. It enters in the dorsal horn. From the dorsal horn, it's going to come out the ventral horn. It's going to go to a muscle, and then I'm going to pull my finger away. That's a response to stimulus. And it's also showing you how the PNS and the CNS are connected. But the next step we had gotten to after that, that kind of made it a little bit more complicated, is, well, there's other places we need to communicate with as well. The cord can't make all the decisions. So a lot of times what ends up happening is I'll send a signal from, say, the body to the cord. From the cord, it goes up to the brain to be processed, and it's going to have to go up along what's called a tract. From the brain, we process that information. It's going to come back down a different tract, and then it's going to exit the cord and then go back out the body. So in that case, the cord is kind of serving as a conduit, a connection between the brain and the body, right? So tracks ended up describing that up and downward communication along the cord, and it would follow different regions of white matter in the cord, right? So again, the difference between gray matter and white matter is the gray matter ended up forming that butterfly formation in the center. The white matter was the surrounding structures on the outside. So gray matter here, white matter here. These are mostly cell bodies that make up gray matter. These are mostly myelinated axons. That's what causes it to be white matter. But then you can subdivide this stuff into different little regions. Like, for example, back here, we had what was called dorsal columns. And dorsal columns were examples of tracts that would carry information up. So they were called ascending tracts, and it would carry a specific type of information. But you also had things like anterior and lateral corticospinal tract, which would carry information downward, right? And if you're going downward, that's a descending tract, and it would carry motor information. But this occurs out here in the white matter of the cord. So what you might see is, like, say, touch and pressure comes in here and goes up dorsal columns. Part of the brain senses that, says, I don't like that, sends a signal back down the cord and back out, and it pulls you away from it. So you could have, again, a much more complicated series of events, you know, as part of this communication. Or if it's something simple, like I'm going to go from the body to the cord right back out, then it's called a reflex, and it's usually a very fast, rapid response. But again, tracks, whether you understand them or not, doesn't mean you're not going to be asked about them. So sometimes when I'm going over terminology with tracks, it's trying to prepare you to how to answer questions about them on a standardized test. Understanding might end up coming much later. So when you get into tracks, there's two main categories. You have ascending versus descending. Ascending is always sensory, descending is always motor, okay? Tracks will always decuss, they'll cross over. So what ends up getting picked up by, say, the body on the right side will end up being processed by the brain on the left. Or if the brain is initiating a signal, it's going to be showing up on the body on the opposite side. So tracks will always decussate. Here's some examples of where they're located. Here's some tracks that are labeled. I pick the big three when we talk about the ascending tracts, usually I end up talking about dorsal columns. First, this is no new information is here other than maybe causes. Lateral spinothalamic tract we talk about. Anterior spinothalamic tract we didn't talk about so much in anatomy 1, but let's just be familiar with it this time, just knowing what it does. And then spinocerebellar. Those are the ascending tracts I typically pick. And then the one that we usually talk about with descending is corticospinal. So again, nothing new here, but what gets asked about them? Things that get asked about tracts is, you know, if you have these ascending tracts carry sensory information up the cord. If I'm dorsal columns, which is made up of fasciculus cuneatus and fasciculus gracilis, right? Again, fasciculus cuneatus picks up information for the upper part of the body. Fasciculus gracilis picks up information from the lower part of the body. When you put them together, it makes up dorsal columns. But what type of information is dorsal columns picking up? Whether it's fasciculus cuneatus or fasciculus gracilis, it picks up light touch, light pressure, and proprioception. So if I'm picking up touch, pressure, and proprioception from the hand, it's going to run along fasciculus cuneatus. If I'm picking up touch, pressure, and proprioception around the knee, then it's going to go up fasciculus gracilis, right? But together, fasciculus cuneatus and fasciculus gracilis make up dorsal columns. It's an ascending tract that carries information about touch, pressure, and proprioception. So proprioception, again, was one of those words. You'll see it again in the next chapter when it gets into neurosensory, but let's just go ahead and define it now, because most of neurosensory, you can just read the slides and there's not a lot of extra description that's needed. But with proprioception, you have muscle stretch, tendon tension, and then joint position or joint angle awareness. I'm just going to put joint pause awareness. So like my finger is bent, that's joint position awareness. Like if my eyes are closed and you say, which finger is bent, and you go and you adjust one of the patient's fingers, he'll say like my fourth finger or my ring finger, right? Which direction is it facing? It's facing toward the ceiling. You know, that's part of proprioception. So but muscle stretch, tendon tension, joint position awareness, that's proprioception. It's basically monitoring where the body is in space without you having to look at it. So, you know, when we're testing dorsal columns on a patient, normally you will tell them to close their eyes because we don't want them to cheat, you know. And you can test dorsal columns a lot of different ways. One way we test it is just by having them say open their hand, and we'll draw something on their hand. You know, maybe I'll draw like a letter, or maybe I'll draw like a shape, you know, something like that. And I'll say, what did I just draw? And if you can tell me, then that meant that the patient had what's called graphesthesia. So aesthesia means to feel, graph means like a picture. So I can feel drawing. But if a person has dorsal columns issues and they can't feel touch or pressure or proprioception very well, then they end up with what's called agraphesthesia. They can't feel drawing on the skin, right? Another test we do is we'll tell them to open up the hand, and we'll stick an object in their hand. And I'm like, and again, their eyes are closed. And I'm like, what object did I just place in your hand? And they're feeling it. And they're like, that's a balloon. And you're like, no, definitely not. You have what's called astereognosis, or you can just call it asterognosis. Oops. There we go. So you can just drop that E sometimes. But stereognosis means you have the knowledge of a three-dimensional object by touch alone. Astereognosis means you do not. So if I told the patient, close their eyes, open your hand, I can place an object in your hand, can you tell me what is this? And they say that's a pen, then that patient has stereognosis. That's normal. So I might not mark anything. But if I say, close your eyes, hold out your hand, and I stick something in your hand, and I say, what is this? And they're like, it's a hamburger. Then I might say, poop, astereognosis. So that could indicate they have a dorsal columns issue. Obviously you want to rule out, that they don't have like a peripheral nerve injury. Like if they have a giant gash in their wrist and the nerve is clearly hanging out, it's probably not a dorsal columns issue, right? But if I can sense touch, maybe somewhere else, and I'm not really able to describe it, but I know something's there, that could indicate that, maybe one of my tracts is working, like my anterior spinothalamic tract is working, because that can pick up crude touch. It doesn't tell me much about the touch, so I wouldn't be able to give you a lot of detail like I could with dorsal columns, but I might notice that there's something there that could tell you that the nerve is still working, right? Or maybe if you poke them and you see that they pull their finger back, that means they can pick up pain, right? So if they can pick up pain, that means the nerve's still working. But we're trying to distinguish whether they can feel light touch, light pressure, and proprioception. And if they can't do that, then that points us more toward a dorsal columns issue. So the more you practice these tests, you get better with it over time. And you'll have, depending on what acuity you work and what floor you work on, you'll have certain tests that the providers like to do, and they'll teach you how to do these tests, but you'll get good at being able to recognize them. So again, with dorsal columns issues, a lot of times patients will experience a graphesthesia, a sterognosis. They're probably going to have balance and gait problems because they have difficulty feeling the bottom of their feet, right? Similar to what we're talking about with distal neuropathy with diabetes, and numbness and tingling. That's pretty common across a lot of neurological conditions. Paresthesia is certainly not just a dorsal columns issue. I mean, paresthesia comes with most neurological conditions, at least early on, but that just means numbness and tingling. Problems that can lead to dorsal columns issues, like what causes lesions along dorsal columns. Diabetes mellitus can do it. Pernicious anemia is one of the big ones that does it. And tertiary syphilis. Lateral spinothalamic tract was one of the ones that we had gone over, and that picks up pain and temperature. So pretty easy to test. I probably showed you in class a long time ago the sharp dull test, which is you could take like a cotton swab, break it in half, and when you do that, you end up having a sharp end and a dull end, because a lot of times in a doctor's office, I'm not talking about the ones you get out of the Q-tip container at home, but in the doctor's office, you'll notice that the cotton swabs have a wooden stick, and then you have the swab at the top. So they're just called cotton swabs, not Q-tips, because it's not the brand Q-tip. So this is the dull end, and then if I break the stick, it leaves a little jaggedy edge. That's your sharp end. So if I want to do a sharp dull test, I can just use a broken stick in the cotton swab. And you are basically looking, can the patient distinguish between something sharp and dull? So if they can feel dull, that indicates that they can pick up touch. But if they can't feel the sharp end, that might mean that something's missing here. Like, I know something's touching my skin, but I'm not picking up pain. So that could be an indicator of a lateral spinothalamic tract lesion. So one of the most common types, hold on, let me share my screen with you again. One of the most common types of lateral spinothalamic tract lesions is something called a syringomyelia, where they have a fluid-filled cyst on the cord. So if I have a syringomyelia, what can I expect in that patient? They have difficulty picking up pain, or usually hot temperature, like burn, pain and burn, pain and temperature. So again, we can do a sharp dull test, and anytime you hit them with a sharp end of something, they'll say dull. Every time you hit them with the dull end of something, they'll say dull, because again, dull doesn't have to be necessarily something around it. Like if I am doing this on my skin here, this has a rounded appearance to it. So of course that's going to feel very dull to me. This isn't necessarily super, super sharp. So that's also going to feel dull to me. But what's going to indicate, so let's say my eyes are closed and I go and I poke it like this. What's going to indicate to me that this is sharp? It's the pain that's associated with it. Because when you make the object sharper, right, it'll be more painful when you hit it up against the skin because it's more likely to cause damage to the skin when it's sharp like that. So again, when a patient gets touched with this end, they're going to say dull because yes, it is a dull surface, but it also gives a dull sensation. But when you hit them with that end, they also say dull because all they're feeling is the compression of the skin. They're not feeling the pain sensation that came with those sharp points, right? They're just feeling the compression of the skin. So hopefully that clears that up if that's confusing. But again, the most common cause of lateral spinothalamic tract issues or problems is a fluid-filled cyst on the middle of a cord called a syringomyelia. Anterior spinothalamic tract, I just want you to be aware of what it picks up. And again, this gets mentioned again in neurosensory. There's different types of touch, and there's different, I guess you could say, degrees of pressure even, in which you have light touch, you have crude touch, you have two-point discrimination touch. And the kind of light touch, fine touch, and discriminative touch are things picked up by dorsal columns. So it's a finer type of touch, right? More detailed. The type of touch that's picked up by anterior spinothalamic tract is more crude, right? So it's, you know, something's touching you, but there's not a lot of detail that goes with it. It's in contact with me, but I can't really describe it. So crude touch. And then a kind of a more dull pressure goes with anterior spinothalamic. So when I think dorsal columns, I think light touch, light pressure, and proprioception. When I think anterior spinothalamic, it's more like crude touch and deep pressure, dull pressure. Let's see. Spinocerebellar, this would carry information to the cerebellum. And if we said that the cerebellum was good at coordinating muscle activity, well, what if the information couldn't get there for me to coordinate? Well, then I would have lack of coordination. So that would lead to a type of ataxia. Let's see. So MS might end up affecting spinocerebellar tracts and the cerebellum. And then when you get into corticospinal tract was good at carrying all your volitional signals or what we call voluntary muscle control signals. So think about the precentral gyrus as being the beginning of this, because the precentral gyrus, when we go back into the brain, was the part of the frontal lobe that was in charge of voluntary muscle activity, or we sometimes call it the primary motor cortex, right? The cortex is just like the outer covering or the outer gray matter of the brain. I don't want to make it confusing, but here's the precentral gyrus. It's in charge of your voluntary motor function. So when it sends signals down from the primary motor cortex, that's where the word cortico is coming from. And then it's going to go down the spine, the spinal cord, and eventually it's going to exit a peripheral nerve and go out to a skeletal muscle. But as it's going from the precentral gyrus down the spinal cord along a tract, it's called corticospinal tract. So that's where that terminology comes from. So that'll help you remember it. And what it's doing, again, is it's carrying voluntary or volitional muscle activity information, eventually, which will exit along a peripheral nerve and help control skeletal muscles. So you can imagine if I had a stroke or a lesion somewhere where these signals are being sent down the cord or in the brain or where they're crossing over, then you're going to have difficulty with bodily movement. So one of my wife's patients not too long ago had what's called locked-in syndrome, which is very rare. But your corticospinal tract, as it goes up, it actually comes up the cord and it crosses over in this kind of region down here. I was trying to find a good picture that had the brainstem on there. But down in this area, and this person most likely had some type of stroke or damage or tumor or lesion somewhere in there, right where the corticospinal tract was going up, and it affected both sides. So what ended up happening is the person was completely aware of what was going on. They can sense most things, they can feel most things, but they have no control of their body whatsoever. They could no longer control their body's muscle activity. So they were kind of locked in their own body, you know. But it was basically they had tumors or they had a stroke right where those fibers of the corticospinal tract were traveling up through the brain stem. So when you get corticospinal tract injuries, depending on the location of the injury, it could create what's called ipsilateral or contralateral, um, sorry, hemiparesis. So let me draw you a picture of that. I think that'll make it easier. When you're dealing with corticospinal tract, let's say the brain's right here. Here's the brain stem. I'm just going to make it a little bit thicker. And then here's the cord. All tracts decussate over at some point in time. So they'll, if it starts over, sorry, let me grab that real fast. This is a high production value video we're doing here. So if it starts over here and it's an ascending tract, it'll end up somewhere over there. Like if I start on, say, the right side of the body, as I go up, if I'm an ascending tract, eventually I'm going to cross over and I'm going to end up on the left side of the brain. So let's start with corticospinal tract over here on the left. As it comes down, it's going to stay ipsilateral in the brain. It's going to get to the brain stem. It's going to cross over here, and then it's going to drop down contralaterally to eventually head out to the body. So if I end up having like a lesion right here, then you're going to have some type of ipsilateral problem with that. But if I end up having a lesion right there, you're going to have contralateral problems. So I did put that in the notes for you because it really just depends, did it happen prior to or after decussation? So when you do get hemiparesis, let's see, if problems occur with the brain, it will affect contralateral. If problems affect in the cord, it'll affect ipsilateral. So from that perspective, I was coming upward. But so just use the notes that I just gave you. But when I was initially drawing the picture, I was coming down. Here it's talking about if this is missing, which part got affected. So we were going up in this direction. So if we end up having a problem right here, is that going to affect this side or is that going to affect this side? That's what that is saying. It's going to affect this side because that's still on the same side. But if I have a problem here and we're coming from this perspective, not from the brain going down to what it works on, but let's say I notice the paralysis is right here, you know, which side would it be on if the lesion is further up? Here it's contralateral, right? So in the brain, it's going to be on the opposite side. If it's in the cord, it's on the same side. So I didn't mean to make that confusing. But when you talk about a corticospinal tract, it actually starts in the cortex and it works its way down the spinal cord. So it actually goes in this direction. But from a, I guess, a clinical perspective, we're not looking at it from its point of origin. We're looking at it from where the deficit started. So if the patient is presenting with paralysis on this side, where could it have come from? If it's a corticospinal tract lesion, well, if it's in the cord itself, it must have been ipsilateral. So I might want to look at a CT somewhere in here. But if it was in the brain, so if this is clear, I would expect it to pop up contralaterally because it had already decussated over. I hope that helps. I hope I didn't make that more confusing. But it's written in clear terms here. Upper and lower motor neuron lesions. Let's keep this one simple again because these are things that are going to come up over and over and over again. Let's say we have a motor problem, because obviously when you're dealing with upper motor neuron lesions or lower motor neuron lesions, clearly something motor has been affected. And when we have, say, motor paralysis, there's two different types. I can have what's called spastic paralysis, or I could have what's called flaccid paralysis. And you can have different degrees. It could be completely stiff and rigid or just partially. It can be completely floppy or just partially. But when you're dealing with motor neuron lesions, there's going to be some effect in how we control our body. And again, if this finger is too weak to be lifted up, I can't use the finger. Or if this finger is too tight and rigid to lift up, I still can't use the finger. So that's a motor problem, right? So let's say I can feel it just fine, no problem. I feel it, but I can't move it the way I want to because it's either too weak or it's too stiff. Okay. So this is kind of the idea behind upper versus lower motor neuron lesions. So imagine a patient gets into an accident, hits their head really hard, and all of a sudden their body seizes up and becomes very spastic in one area. That's an example of an upper motor neuron lesion, right? Or imagine somebody has a impingement of a nerve somewhere in the periphery, and their body becomes weaker and weaker and weaker, everything distal to that nerve. Eventually, it might even atrophy and become smaller. That's an example of a lower motor neuron lesion. So how do I tell the difference between upper motor neuron lesions versus lower motor neuron lesions? And what is, I guess, the root cause of the way it appears? And what's the underlying mechanism? So if somebody has an upper motor neuron lesion, chances are normal movements are going to be replaced with more spasticity. So the muscle becomes more spastic. So with upper motor neuron lesions, rather than having normal movements, their muscles have become more rigid or more spastic, right? So they might be difficult to move because they're stuck. If you do any type of reflex test with them, like deep tendon reflexes, the reflexes are elevated. So we say they're hyperreflexic, hyperreflexia. And you might even notice that they have certain pathological reflexes present. So again, you'll probably learn these when you're on the floor. But some examples of a pathological reflex is if I tap somebody on the fingernail and their hand kind of clenches down like this. Or if you stroke the bottom of the foot real hard with a sharp object, their toes flail. That's called a Babinski response, right? So Babinski is an example of a pathological reflex. So if you have presence of pathological reflexes, spasticity, and hyperreflexia when you're doing deep tendon reflexes, those are all good indicators that this person has an upper motor neuron lesion. So what does that indicate to me, and why is it occurring that way? That probably means the problem is somewhere, again, here, 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 right? It's somewhere in the brain or the cord. If you want to know the specific cutoff point for an upper motor neuron lesion, if we go all the way down to the cord, so remember, corticospinal tract starts in the precentral gyrus. It's going to go down through the brainstem, decussate over. It's going to go down the cord. But then eventually it needs to communicate with, so let's just say it's going to communicate with one of the cell bodies in here. From here, it needs to exit out and go to the skeletal muscle. This guy right here is an alpha motor neuron. That's what it's called. It's called an alpha motor neuron. Sorry about that. It's called an alpha motor neuron, but it's also your lower motor neuron. Your upper motor neuron was up in the precentral gyrus. This is your lower motor neuron. So upper motor neuron lesions can occur anywhere between the precentral gyrus and right here. Lower motor neuron lesions can occur from here out. So usually a lower motor neuron lesion is a peripheral nerve injury. This is the most likely place where that's going to be affected, right? Upper motor neuron lesions usually occur from some type of stroke or trauma, right? So the easy way I tell people to remember that is if you have an upper motor neuron lesion, the lesion is somewhere in the brain or the cord. It's somewhere in the brain or the cord. probably coming from stroke or trauma. And again, I know lower motor neuron lesion, it starts in the cord, but it's right at that edge of that ventral horn. So most of the time, it's peripheral nerve injury. It's where it's already exited, because that's the more likely place it's going to get affected. So when you get into lower motor neuron lesions, let me just flip this over. Lower motor neuron lesions are the exact opposite. Typically, we are flaccid rather than spastic, so it's weak. Like you would tell the patient, like squeeze my hand or something like this. And let's say we have carpal tunnel syndrome. Carpal tunnel syndrome, your median nerve gets compressed right here. So this muscle starts to atrophy and the ability to adduct the thumb goes down. So they would have very difficult time squeezing, right? They'd be very weak. The muscle would be very flaccid. Eventually, it leads to muscular atrophy, or what's called denervation atrophy. The muscle starts to disappear, but the muscle is flaccid and weak, and they become what's called hyporeflexic. And if you ever get asked, what scale do we use to measure reflexes? It's called Wechsler's scale, and I'm pretty sure that's spelled this way. There might be a silent H in there somewhere, but it's called Wechsler's scale of deep tendon reflexes. I might have just made that up, but I don't think I did. I'm pretty sure that's right. And then the other thing I told you about in Anatomy 1, sometimes they'll ask, what are examples of diseases that are upper or lower motor neuron lesions? ALS is the one that's both. It has both signs and symptoms of upper and lower motor neuron lesions. Polio used to be the most common form of lower motor neuron lesion. Guillain-Barré is more prevalent now. We'll talk about that later. This actually might be polio. This actually might be a version of polio. And then when you talk about upper motor neuron lesions, again, usually you think about stroke, trauma, or maybe prolonged demyelinization that's caused lots of lesions. So MS could even be an example of an upper motor neuron lesion. And then lastly, I think the only other thing I really skipped over, again, you can always ask me questions about anything once I get back. I went past seizures, did I not? Let's see. So when you get into seizures, I think the thing I want to talk about these the most with you, most of the time you don't have to determine why this person has a seizure. I'm trying to say that carefully. It'll usually come, let's just say it this way. It'll usually come from one or two things in your practice, in your environment. And it's usually an electrolyte imbalance, or it could be febrile. So it could be coming from a fever. So that's probably, in my estimation, the most common time you're going to see seizures, and you're going to be treating those seizures. Because for the most part, people that have like epilepsy or people that experience seizures, they're random. There's not a lot of treatment that we do on site other than keep them alive at the most basic level. But if they're having no, say, normal history of seizures, and all of a sudden they're coming into the hospital and now they're acquiring seizures, then chances are, what changed immediately or acutely between 5 hours from now and just now, maybe they're spiking fever, so it's febrile seizures, or maybe there's an electrolyte imbalance. That's probably the two things I would look for. And that would be the kind of the things that you would be mostly responsible for correcting. When you start to get into the grand mal, petit mal seizures and all the different classifications, these just end up being more board type questions, right? So when you get into partial onset seizures, it arises from a single focal point of the brain. So you might end up seeing a small area of the body affected. But typically what ends up happening is you get almost like this lightning bolt kind of progression where things tend to spread out, and it can kind of start in one little area and progress to other larger areas. With a simple partial seizure, though, consciousness is preserved, so the person is pretty aware. But maybe they're just stuck in a position. Maybe they're just they're able to talk to you, they're able to converse with you, but part of their body has become rigid. Doesn't necessarily have to shake or anything like that. But there's something wrong. There's something inhibiting them from their normal function. But in this case, with a simple partial seizure, they're completely communicating with you, completely aware. When you get complex partial seizures, there will be in and out consciousness. The patient themselves might not shake necessarily, but you might notice that you were conversing with them and all of a sudden they're looking at the ceiling. They're not really paying attention to you anymore. Maybe the eyes will shutter a little bit. Like you might notice that they don't necessarily roll up, but when I see this in patients, what they sometimes do is they'll kind of tilt back a little bit and they'll repeat that a couple of times. But it's almost like they're looking at some type of fixed position above you. And in your mind, you're like, I'm right here. What are you looking at? But again, the more you see it, you're like, oh, he's having a seizure right now. So then they might start smacking the lips. The eyes might start moving a little bit, blinking. If you're really lucky, there's going to be screaming and shouting. Right? Those are probably going to be the good nights for you, right? But at least those nights will kind of go fast and you'll have something to talk about later. When you get into the more generalized seizures, so as they progress and they tend to spread out, you might end up having the kind that you usually think about when you think about seizures, where the body is writhing and shaking and convulsing. So in public, what's the treatment of that? Just make sure they're in a nice, safe space, right? Let's see. What else did we want to talk about, if anything? I can't think. Oh, I think there was one other thing I'd looked at, and I said I did want to mention this later. I'm not going to test you over this, just in case you're wondering. So I did want to talk about this, but dermatome mapping is a fairly useful thing. When you're learning about neuro, there's so much we could literally spend years on it, and it would still be hard to encapsulate all the stuff that we could learn from it. But a lot of understanding neuro is when I understand basic function and I understand who's responsible for things, I can kind of make a logical or educated guess that when something's absent or it's not working appropriately, I can find a focal point in the brain that might be responsible for that, or maybe I know a pathway that might be responsible for that. So when you get out into peripheral nerve problems versus what's called nerve root problems, it gets pretty fuzzy. Because when you get into things like, say, brachial plexus and you get into lumbosacral plexus, what ends up happening is you get combinations. So for example, let's say we have like a C5 spinal nerve, we have a C6 spinal nerve, we have a C7, C8, and T1. When you go on to form things like brachial plexus, these end up like conjoining together and they end up forming branches and all this other kind of stuff where you have multiple nerve roots that come together. And when they branch back out again, you end up getting more than what you started with. So you end up getting things like muscocutaneous nerves, and you get radial nerves, ulnar nerves, median nerves. And so it becomes more complicated than just kind of understanding, well, where did it stop and where did it finish? Because sometimes along the way, we end up conjoining and rediverging again. So sometimes in neuro, what we end up having to do is we kind of have to separate, well, what if somebody has a nerve root impingement, which usually comes from disc injuries or bone spur injuries, versus a peripheral nerve injury, because there's a lot of overlap in these things. So let me give you an example. Let's say a person came in and they experienced a problem here, like this part of my hand and maybe this part of my wrist. Well, that could be one of two things. That could be the ulnar nerve, or maybe that could be my C8 nerve root, right? And the way I'm determining that, the reason I'm telling you that's what those are, is because when you look at this dermatome chart, and I know this is hard to see, C8 would be represented by that little green patch right here. Same thing on this other side. You see that C8 right there? C7 tends to be more of the middle of the hand right here. C7 tends to be, or sorry, so C8, C7, C6 right here. So C7 is kind of in the middle of the hand, front and back. C8 tends to be over on this side. C6 tends to be right here. So when somebody says this part of my hand's numb, well, there's two different things that could affect that. It could be the ulnar nerve itself, because a lot of times people get damage to the ulnar nerve right here. And that would affect this part of the hand. Or it could be a C8 nerve root impingement. So how could I tell the difference? Well, one thing I could do is I could think, well, what controls muscles in a certain region? So we have what's called dermatome charts that will help me to localize nerve root issues. Like dermatome charts follow nerve root areas of skin that say a particular nerve root is responsible for. So let me blow this up so you can see it a little bit better. For example, my C3, let's say my C3 nerve root is responsible for this little area around the throat or the neck. My C4, the upper part of the shoulders. When you get into C5, it starts to be a little bit lower on the shoulders, and it goes all the way down into the upper lateral portion of the arm and the upper lateral portion of the forearm. What part of the nervous system is picking that information up or is responsible for that information? It's the C5 nerve root. So when you get into a dermatome chart, dermatomes are talking about a part of the nerve, like for example, if this is a spinal cord cross section, and you have nerve root, and then you end up having peripheral nerve. This is a nerve root. And the reason nerve roots sometimes get affected, so it could be here or here, is oftentimes due to bone spurs that happen to be around this area of the spinal cord, or discs, which happen to be in between the vertebra that are surrounding this little area. So if you want to know where this is located in relationship to, say, the vertebra, here's a vertebra right here. Here's pedicle, lamina. Here's the back of the vertebral body. Sorry about that. Here. The spinal cord is pretty small. It sits in this little area right there. So that's the spinal cord. So in between each vertebra, you have discs, and sometimes the disc can bulge out. Like you'll get a disc bulge here. Sometimes you'll get a disc bulge there. Sometimes you'll get a disc bulge there. Every once in a while, you can get one in the middle, but it's pretty unlikely. So here in this little area, that's where these little nerve roots would have came out, and they can be impinged. So if I impinge that nerve root, a lot of times that'll show up in an area of the skin that'll feel numb, or it won't feel the same that it used to. So if I have impingement of a nerve root, we sometimes use our dermatome chart, and I'll just go along and I'll feel the skin on the patient. You can see where I've marked myself with pen the whole day. And I'll ask them, I'll say, how does this go? Can you feel me touching your skin right there? And they're like, yes. So in my mind, I'm like, okay, well, C6 is good. How about here? Can you feel this? And they're like, yes. And I'm like, okay, well, C7 is good as well, because that's the area of the body, or at least the skin on the body, that my C6 nerve root is receiving information from. This, my C7 nerve root is receiving information from. Here, my C8 nerve root is receiving information from. So sometimes, if a patient comes in and they say, I have numbness right here, well, it could be coming from two things. It could be coming from a nerve root impingement, or it could be coming from a peripheral nerve problem. And sometimes we have to use multiple types of information. So if a patient came in and they said they had pain right here, well, I've narrowed it down to at least two things. We know it's not the C7 nerve root. T1 would be further up. So I know it's either the C8 nerve root, or I know it's the ulnar peripheral nerve, right? And so the way we can kind of determine is we can go and use another chart. And what the other chart would show us is what's called myotomes. And myotomes are usually things that are represented by peripheral nerves. So for example, if I'm talking about the ulnar nerve, let's see, usually and this isn't the chart I was actually wanting to use, but it kind of works. What we end up seeing is an ability to say, bring this finger in, so we can do adduction, and some of this musculature works. And then you can also get what's called ulnar deviation. So what I could do, and again, that wasn't the best chart to show that on, is I could test for muscle strength or muscle weakness. And I would do that bilaterally from one side to another. So that might be a way that we could say, well, I think it could be C8, or I think it could be the peripheral nerve. But let's say that the muscle tone was preserved, which ulnar nerve would do, but it was just the skin region that was affected. Then I could eliminate it wasn't the peripheral nerve because the motor function was preserved. So we know that wasn't affected, but the dermatome was. So I'm not going to try to make this sound like it's easy by any stretch of the imagination. To me, one of the hardest things you can possibly do in neuro when it comes down to those little finite areas is determine whether something is a nerve root injury versus a peripheral nerve injury. But again, that's why we have neurologists on staff. I don't think it's something you would ever be responsible for, but that's the use. Let me go back to this picture. Sorry. But that's the use of charts like this, just in case you're ever wondering. So I'm never going to make you memorize this chart. I'm not going to make you memorize this chart, but I did want to kind of tell you how we end up using those things. Like sometimes we'll use these things to determine nerve root impingement syndromes, maybe peripheral nerve injuries. And the charts I gave you were actually both nerve roots. I thought I gave you a peripheral nerve chart, but I didn't. But that's what those get used for. So what I'm telling you is you don't have to memorize these. I was just showing you how they get applied. Shingles, there's another one. Shingles is one of those things that you will probably run into quite a bit. One of the herpes viruses, the thing that kind of stands out about shingles to the patient is they're very painful blisters. So they'll usually, well, the terminology given on a test, maybe the patient themselves won't actually use these words, but like hot poker type of pain. Like if you took like a hot poker in a fireplace and then you stuck it to the skin, that's the type of pain that comes from shingles. The word we use to describe the blisters is called vesicles. So if they use the word vesicles and they say the vesicles are following a dermatome, then you can always assume that they are talking about shingles. So shingles is kind of a sequela of a herpes virus from when you had chickenpox as a child. We sequestered it and then somehow it escaped. So it's almost like a jailbreak of chickenpox. But a lot of times it'll happen to people in the hospitals because they're already under a lot of physical or immunological stress. And the immune system takes a lot of energy to work optimally. And if it's stressed out by other physical ailments or trauma, sometimes we don't have the resources that we used to kind of keep other things in check, So they might get these side infections, or they might get things like shingles that all of a sudden pop out. But if you ever want to identify shingles, hot poker-like sensation of pain is the words they use to describe it on a standardized test. The type of blisters are called vesicles, and the vesicles always follow a dermatome, right? So usually it'll be on one side of the body or the other, and it'll be in a stripe-like pattern. So that's the other place where you might use the word dermatome. But that should do it for this presentation. So what was that, like 15 minutes, 20 minutes? So no problem. I'm sure you got this. A little bit later today or this weekend, I'm going to do a video for you of neurosensory. It's going to be very short. Most of it, you're just learning and reading different types of receptors. What are they called? How are they classified? It gets back into special senses exactly the way it was before. What does the retina do? What does the cochlea do? What are different parts of the ear? So I think it'll be fairly brief, but we will go over cranial nerves again. So I'll go over the cranial nerve chart with you. But that's it for this one. I'll post this one in just a little bit.